Hello, everyone, and welcome to today's symposium. I'm Charlene Lancaster, Assistant Science Editor for The Scientist, and I'll be moderating our discussion. Today, our expert panel will explore the expanding potential of adeno-associated virus technology and its role in advancing cell and gene therapies. Before we begin, I'd like to thank our symposium sponsors: Vector Biolabs, Shimatsu, Takara, Sino-Biological, Beckman Coulter Life Sciences, Biorad, Taconic Biosciences, PHCBI, BPS Bioscience, AdGene, Innovative, and Mission Bio. We like our symposia to be interactive. We encourage you to send us your questions or comments at any point, and our speakers will address these during the Q&A sessions following their presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. Our sponsors have provided us with some helpful resources related to today's symposium, and we've posted these in our handout section on the right side of your screen. You can access and download these documents at any time during the symposium. If you've minimized the Q&A panel, simply click on the blue chat button in the bottom right corner to bring it back. This symposium will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. And with that, let me introduce our first speaker. Bill Legore is a professor in the Department of Integrative Physiology at Baylor College of Medicine. He completed his PhD in biochemistry at the University of South Florida College of Medicine and a postdoctoral fellowship in the Division of Translational Medicine and Human Genetics at the University of Pennsylvania. Legore is an expert in lipid and lipoprotein metabolism and gene therapy for metabolic diseases. Understanding and improving AAV gene therapy is a major focus of Legore's research. His laboratory is developing new gene editing approaches focused primarily on the liver and the vasculature. Let's make sure your slides are up and running. Okay, Bill, take it away.
Hello, everyone. Today I'm going to talk to you about AAV tropism in preclinical models. My name is Bill Legore, and I'm a professor in the physiology department here at Baylor College of Medicine in Houston, Texas. So just a little bit of background, because I realize we probably have different folks in our audience with varying levels of background about adeno-associated virus, or AAV. This is what AAV looks like in terms of the wild-type virus. It's one of the smallest viruses in existence. It has a non-enveloped protein capsid surrounding a single-stranded DNA genome. In and of itself, it cannot complete its own viral life cycle, so it needs actually a helper virus for replication. It's a remarkably efficient virus in that it has only two genes, rep and cap, which give rise to a number of different protein products shown below. For the purposes of this talk, we're going to focus mostly on the cap gene, which actually encodes the capsid, the protein shell of the virus, which is really important in determining which cell types and tissues the AAV virus can actually get into. Okay, so when we make recombinant adeno-associated virus, this is a really nice system where basically we can introduce any new genetic material that we would like into the AAV capsid, provided that this sequence is flanked on either side by these short elements called inverted terminal repeats, or ITRs. And as long as we have those ITRs, we can package pretty much any sequence we want, say a tissue-specific promoter and your favorite gene, provided it fits within the relatively small packaging limit of AAV, which is about 4.8 kilobases from the beginning of one ITR to the end of the other. So AAV has definitely proven itself to be a very effective gene delivery system, both for basic science applications as well as for human gene therapy. AAV has been used in over 250 clinical trials to date, and at the present time, there are already 7 FDA-approved AAV gene therapy products in the US that are actually being given to patients. So pretty remarkable time to be able to actually fix genetic diseases with these engineered viruses. Okay, so just a basic primer on what the AAV capsid looks like. So the AAV capsid is composed of these products of the cap gene viral protein 1, 2, and 3, VP1, VP2, and VP3. So these VP subunits come together in a roughly 1 to 1 to 10 molar ratio to form a 60 subunit capsid. Okay, so this is basically what it looks like down below. By mass, most of the subunit is VP3, but it's important to note that these VP subunits, although they're varying in length, they can all connect with one another. So they essentially are interlocking Lego pieces that are more or less interchangeable. And what you'll notice also is that many of the important structural features of the virus are actually going to reside at interfaces between these different VP subunits, which we'll get into in just a few minutes. Okay, and as I mentioned before, although they are different sizes, beginning at the N termini, they have the same C-terminal N, these VP subunits, and the way they come together to form the capsid is actually fairly random. It actually assembles in a stochastic fashion so that they can all interchange for one another in different positions. They're pretty much perfectly interlocking and interchangeable in terms of what is exposed on the surface of the capsid. This is a nice paper by Warner et al. In Nature Communications that used the mass spec approach to basically quantify the distribution of the different VP subunits within recombinant AAV capsids. And this is kind of what the AAV capsid looks like on the right in terms of its crystal structure. So it gives you a little bit better idea that it actually does have some meaningful shape to it. And you can see these different features. You have the fivefold axis, and it has this depression surrounded, the circular depression surrounding this fivefold symmetry axis. You have these threefold protrusions at the threefold symmetry axis, which are really important in antibody recognition, as well as receptor binding. But this just gives you a picture of what the virus actually looks like, in the real world, at the atomic level. Okay. I'm just going to talk a little bit about some important features of the virus. As I mentioned, that threefold symmetry axis, if we just take 3 VP subunits and make that up, we can show you a trimer. Here, this on the left side is AAV4. On the right side is AAV9. Each subunit is a different color of either white or gray. In the case of AAV4, we've highlighted residues that have been determined by others to be important for binding to the specific sugar that AAV4 binds to. That's alpha 2,3-O-link sialic acid. And then we have the same thing for AAV9, where AAV9, this pink area, is actually the binding pocket for galactose. Okay, so the important thing to know about AAV is that the first interaction with proteins on the cell surface is really mediated by carbohydrates for the most part. So they have these glycan binding motifs to interact with sugars, which may be present on many different proteins. And this is their first interaction with the cell surface before they can be endocytosed. And so important thing to know about this is that you can see the AAV capsid is very complex. And so this really can present a major challenge for engineering new tropism for the virus, or engineering it to bind to new receptors, in that you don't have exactly a linear molecule to deal with. A lot of these receptor binding interactions actually exist across multiple subunits, and if you make one change here, you have to make another change somewhere else. So it could be a very challenging problem for protein engineering. Okay, as it turns out, there are many different flavors of AAV naturally occurring. So this is really a landmark paper by Guangpeng Gao when he was at Penn, where he cloned a whole wide range of naturally occurring AAV serotypes. So AAV were initially defined by serotype based on their antibody, based on which antibodies can recognize them. And so most of us have heard of AAV1 through 9 as kind of the most commonly studied naturally occurring serotypes. But AAV can also be grouped into clades based on the sequence of the cap gene that makes up this VP subunit that we were just talking about. And you can see on the left here, this phylogenetic diagram shows you kind of the different clades of AAV where, more closely related serotypes are grouped together. The HU designation means a human isolate. The RH means a rhesus isolate. And we also have AAV from a number of other mammalian species out there as well, if people are interested. Important thing is that when we make recombinant AAV, the nice thing is that we can use the cap sequence from pretty much any of these naturally occurring isolates and use that to make a recombinant virus. And by exploring new serotypes that exist in nature, hopefully this will help us improve the range of targetable tissues, and in some cases, perhaps help us avoid neutralizing antibodies to the virus. Okay, just the very basics of AAV transduction at sort of a bird's eye view. What I'm going to refer to throughout this talk as functional transduction is essentially the entire process of the virus entering cells, delivering its cargo to the nucleus, and then expressing its genes. And in the case of AAV, what happens is we have this initial interaction with carbohydrates on proteins on the cell surface. So you have this initial glycan interaction, and then you'll interact with an endocytic receptor that actually brings the AAV variant into the cell. It then gets trafficked to the endosome and then has to escape the endosome. And there's actually special domains in the AAV capsid that mediate this process through a phospholipase activity. Then the entire AAV capsid, what survives proteolytic degradation, is trafficked to the nucleus where the protein shell uncoats, releasing the single-stranded DNA genome, which then inside the cell nucleus is converted into these circular double-stranded forms that we call episomes. And so these episomes are sort of the long-term persistent, functional unit of the virus that remains inside cells and can continue to express either viral proteins or transgene proteins, much like a plasmid would if you were to do a plasmid transfection. And so all of these steps together are, cumulatively required for you to get functional transduction. That is, take up of the virus by the cell all the way to the point at which the transgene cargo is expressed. Okay, and so what mediates uptake of AAV? There's been a lot of work over the years looking at least at most of the common naturally occurring AAV serotypes, and there's been numerous receptors that have been discovered. In some cases, there's specific proteins. In some cases, there's more than one protein that can be a receptor for a particular AAV serotype. But what I want you to pay attention to here in this fourth column, where it says primary receptor, is the glycan affinity of the different AAV serotypes. So what you'll notice right away is that a lot of these AAV serotypes all bind to sugars, but slightly different sugars. For example, AAV9 binds to N-link galactose, AAV4 binds to alpha-2,3-O-link sialic acid, AAV2 binds to heparan sulfate, et cetera, et cetera. And so this initial interaction with sugars on the cell surface is extremely important, meaning that these AAV actually can bind to many different places, and just depending on what's expressed on the surface of your cell type and your tissue in a particular species has a big impact on where the virus goes. Okay, we would be remiss not to tell you about AAVR, which is the AAV receptor, which was only discovered in 2016 in really a beautiful paper by Pillay et al. I really like this paper. This is one of the first real good examples of using a genome-wide CRISPR screen to identify a receptor for viral entry. They did a fantastic job of it. They found this protein KIAA0319L, which is sort of uncharacterized at the time, now known as AAVR. This is a plasma membrane and Golgi protein that's capable of endocytosis. It binds to most, but not all AAV serotypes through these repeating polycystic kidney disease, PKD domains. And so most AAV actually get into the cell through use of the AAVR after that initial glycan interaction. However, there are, of course, some exceptions. AAV4, AAVRH3233 are two notable exceptions that do not use AAVR. Okay, and then there is this whole field that some of us are aware of called capsid engineering. And really the goal of capsid engineering generally is to either change the natural tropism of the virus in terms of where it goes to deliver its cargo to new cells or new tissues, and or avoid neutralizing antibodies or immune responses to epitopes on the capsid. Okay, and there's many different ways this can be done. There's very conserved regions of the AAV capsid, and then there's these variable regions, which typically are the ones that are most accessible to receptors, where you can make, perhaps more precise changes. There's many different approaches for diversifying the capsids, ranging from PCR shuffling to domain swapping, chemical attachment, site-directed immunogenesis, et cetera. And with the advent of next generation sequencing, you can make really nice libraries and do your screens in cells or in vivo or even in multiple species, which really gives you a lot of bang for your buck in terms of the design space that you're able to query. And so this brings us to kind of the setup for the motivation for our work. I'm calling this the three-body problem with AAV gene therapy. It's actually a great Netflix science fiction series if you're interested, but I like this kind of title that, typically if we're developing a new gene therapy, almost always it starts in a mouse model because they're very easy to manipulate genetically. They're great to show, that you have a disease phenotype and that it can be corrected in the right tissue by re-expressing the transgene that you're missing. So things almost always start in mice. Typically after that, you'll need to progress to a larger animal model, in many cases non-human primates, to establish the safety of your approach before you can do a clinical trial in humans. And really, the three-body problem is that mice are not humans, humans are not macaques, et cetera. And so what works well in a mouse doesn't necessarily mean it's going to work well in a macaque. And even if it works well in a macaque, it doesn't necessarily mean it's going to be safe and effective in a human. And so we have this question about how do we go from one model organism to another and be, assured that AAV is going to perform as we intended to. Okay, and so I'm just going to give you like three very simple examples from literature that I think illustrates the importance of considering AAV tropism in the context of different species. The first one is trying to get AAV across the blood-brain barrier following systemic delivery. So ideally, if you want to do a gene therapy for a neurological disorder, you don't want to go drilling holes in people's heads if you can help it. So it's long been a goal to give an intravenous administration of the virus and get an AAV capsid that can get you across the blood-brain barrier and into neurons and other neuronal subtypes, cell types. So this is some really nice work that Ben Deverman did when he was in Viviana's lab. And basically what he did was made a capsid library that consisted of a small peptide inserted at a specific position in an exposed loop of AAV9. And he took this AAV capsid library, did some in vivo selection in a Cree-transgenic mouse using a very clever approach that would tell them that this capsid was being recombined and the reporter was being turned on, basically only in neurons, showing that it had gotten across the blood-brain barrier. In doing so, he identified AAV PHPB as well as PHPEB, these peptide insertion mutants of AAV9 that get across the blood-brain barrier very well in mouse models. It turns out the receptor for this is this Ly6A protein. Down here on the left, I'm just showing you what it looks like. Really nice GFP transduction of the entire mouse brain. But the problem that quickly became evident was that this works really well in black 6 mice and a couple of other strains of inbred mice, but it doesn't work in all. So BALB-C, for example, is a mouse strain that is not permissive to AAV transduction with this particular engineered capsid serotype. And so that really is a problem, that we have a capsid that works great, not only just in one species, but in one specific strain of one species. It's probably not going to be translatable to humans, right? And so some really nice follow-up work by Ben's lab. They've really taken this lessons learned from this PHPB variant and designed some pretty clever approaches to intentionally target receptors that would be expressed on the human blood-brain barrier using the same kind of library, next generation sequencing approaches. And they've done a really nice job of identifying the transferrin receptor as a great receptor for AAV transduction of human brain and something that's going to probably be translatable. And so they have a nice knock-in model where you can actually look at how well these capsids get across mouse models that express this human receptor. So this is just an example of how the species you're working in is important, and not only the species, but in some cases even the specific strain does not mean it's going to translate all the way to humans. So how do you get around this problem? This is an approach taken by Bev Davidson's group. There's many others doing similar kinds of things, both in academia and industry. They're saying, let's just get as close as we possibly can to the human situation. Let's go do our screening in a non-human primate model. And so you can go into a non-human primate model. In this case, they're going in there with an AAV peptide insertion library and do several rounds of selection through next generation sequencing to identify capsids that do a really good job of getting across the non-human primate blood-brain barrier and into neurons. And so through this screening, they've identified some really nice candidates that probably are more likely to be translated from macaque to human than if you were, say, trying to go directly from mouse to human. So I think it's a very valid approach. It's an expensive approach, but it's one being pursued by a number of labs, a number of industrial groups, and is definitely bearing some fruit. So we also have the problem though of reverse translation, and that a a lot of the gene therapies, the initial proof of concept that you can actually correct a genetic disorder really begins in a mouse model. It really always begins in a knockout mouse model of the disease. It's often not practical to have a knockout non-human primate version that actually has the pathology that you're trying to correct. And so we have this concept of reverse translation. So not only do we need to be able to go from the capsid that we've designed or tested in macaques or other monkeys to human, but we also have to be able to go back to a mouse model and have something that's going to work there. And so this concept of making cross-compatible AAV capsids, I think, has a lot of value. So there's some work by Aravind Asakan's group where they did directed evolution by basically putting an AAV capsid library through multiple species. In this case, pigs, mice, and then non-human primates through several rounds of evolution to identify serotypes that are more efficient at delivering their cargo across species than the parental, naturally occurring AAV serotype. So I think this approach is also incredibly useful, especially for those of us that work in mice as their preclinical model. Okay, so getting back to this three-body problem that I mentioned earlier, one of the things that we recognize that we really are lacking is a deeper knowledge of AAV tropism in mice, which is where most people begin. Mice are still the main preclinical model for assessing pathogenicity of variants as well as disease correction. And unfortunately, there's really very few systematic comparisons of tropism, even in mice. And so this is basically a huge team effort that came about through the Somatic Cell Genome Editing Consortium. We were part of phase 1 of the SCGE, which was a very large NIH-funded initiative to improve genome editing delivery and technologies. And this was a group, a collaborative effort between 3 groups as part of the SCGE. That was Guangping Gao's group at UMass Med, Jason Heaney and my group at Baylor College of Medicine, as well as Steve Murray's group over at the Jackson Labs. And we had written a supplement to our grants as part of this consortium to actually directly address this question. What is the best naturally occurring capsids for which tissues in mice? And we divided the responsibilities. Guangping Gao's group did a great job designing and packaging all the viruses. The Baylor College of Medicine team did a lot of work on the reporter validation and the biodistribution of the vectors, and then a lot of the fluorescent imaging I'm going to show you later, as well as looking at HSPC transduction was done at Jackson Labs. Okay, and a lot of our inspiration for this work came from this, I would say, seminal paper by Carmela Zincarelli that was published in Molecular Therapy in 2008. And what they did was actually quite simple. They looked at AAV serotypes 1 through 9 in mice using a luciferase reporter to see basically where the viruses went, both in terms of biodistribution, how many vector genomes, per tissue, as well as what they look like in the mice. And I really like this paper. This was sort of one of the first uses of in vivo luciferase imaging. Now it's commonly used, especially in the cancer xenograft space, but this image on the far right definitely drives home the point that many of these naturally occurring AAV serotypes can give you very prolonged expression in mouse models, and a lot of that really targets the liver. So most of these hit the liver pretty hard, and then after you sort of fill up the liver, you'll start to transduce other tissues. So this was really a nice paper. We like this a lot. We use this to sort of model our studies and figure out how can we go bigger and better than this to really expand our knowledge of where AAV goes in mice. Okay, and so this is our paper that we recently just had accepted to molecular therapy called the Comprehensive Atlas of AAV Tropism in the Mouse. And really what we tried to do here was we took different naturally occurring AAV serotypes and we injected them into reporter mice, and we look at both biodistribution by tissue as well as which cells in each tissue are being hit based on fluorescence. So first thing we had to do was decide which AAV serotypes made the most sense to test. We felt that AAV2, there had been a lot of work on it, and there wasn't going to be a whole lot of new knowledge gained by working with AAV2. But aside from that, we tried to pick naturally occurring serotypes that were pretty diverse, fairly well separated in terms of their phylogeny, so that we get some better information about where these go and what that means for actual capsid biology. And so these are the ones we selected, shown here on the left, and you can see a lot of them are very similar on the bottom in terms of their amino acid sequence, with AV4 and AV5 being the most divergent of the group. So how would we detect AAV transduction in mice? Dan Wang and Guangping Gao came up with this very clever way to read out, AAV transduction in a single vector. They use a bidirectional promoter that's ubiquitously expressed. In one direction expresses Cre recombinase, and the other it directly expresses this ZS green fluorescent protein. And the concept here is that if we go into a reporter mouse, such as the AI9 allele, where we have a lock-stop-locks cassette that can be recombined by Cre, this would be a permanent mark where if you had Cre activity, you would then permanently express tomato in all of the daughter cells. And so any cell expressing this red tomato fluorescence, would be permanently red. So this is sort of a very bright binary on or off signal. If you've ever seen AAV or any of the parental cells that led to you has seen AAV, you'll be red. But at the same time, we can also look directly at ZS green, which is active expression from the virus itself. And so these really give you different readouts of transduction. So the ZS green you can sort of think of as a dimmer switch. Some cells might get 1 copy of virus, some cells might get 20, some cells might get 100. So you have sort of this range of expression that you can get in terms of ZS green fluorescence that gives you some information about how much got in there. Whereas the TD tomato signal is sort of you were here at one time, and then we recombine the allele, and now it's permanently red. So this is sort of a binary on and off that I think you're going to see later is really advantageous in terms of very clearly discriminating real signal from background fluorescence. Okay, this was our experimental design. We took these AAV vectors that were made by Guangping and Dan at UMass. We looked at AAV serotype 3b, 4, 5, 6, 7, 8, 9, RH8, RH10, and RH74. We gave a relatively high dose of 10 to the 12th genome copies per mouse so that we would fill up the liver and have an opportunity to look at peripheral tissues actually getting transduced. We did this in the AI9 reporter mice, in this case homozygous AI9 mice. And then 4 weeks later, we collected 22 different organs shown here to look at biodistribution by qPCR, as well as functional transduction by fluorescence imaging. And so this was split up between the Baylor group and the JAX group, but essentially completing exactly the same experiment in parallel. Okay, if we look very briefly at biodistribution, this is all of the different serotypes on the top here, and you can see all of your organs. Here on the left side, which you'll notice right away in the heat map, is that there's a huge bright signal for liver. This is expected. Most AAV serotypes love the liver. The liver has a very porous, sort of leaky endothelial barrier, and a lot of things get in there very easily. There are some exceptions. For example, AAV3b and AAV4 don't transduce liver very well. AAV4, as it turns out, the highest tissue transduced was lung. This is something that Zincarelli also saw in their molecular therapy paper, which we're going to come back to later. But this is basically a broad picture of what it looks like in terms of where the virus is going. This doesn't necessarily tell you that the transgene cargo is being expressed. It just tells you that the AAV genome did get into these tissues. And so if you're a researcher who is studying a particular tissue, say you're interested in lung, you might want to ask the question, okay, which of these naturally occurring serotypes is best for getting my gene of interest into the lung in mice? And so you can kind of slice the data up this way and graph it by tissue, and you can look at all the different serotypes, and you can tell right away that AAV4 is a great choice for lung. Whereas if you like to study liver, like my lab often does, there's many different options that are quite good, including AAV8 and AAV9, which are arguably the most frequently used to target liver. And we also can look at sex differences, and there's a bit more discussion about that in the paper as well. Okay, and one of the questions we wanted to ask right away was which reporter method is actually better? So I'm going to give credit to Michael Berry's group, who a number of years ago in human gene therapy came up with this sort of, they call it Cre fingerprinting approach to detect viral transduction, which is essentially what we're doing here. And they did it with luciferase as well as with fluorescent proteins, basically showing that this Cre activatable transduction readout really makes a lot of sense. And then this was looked at in a little bit more detail recently by Bev Davidson, where they actually compared the direct fluorescence readout to the Cree activatable reporter. And in their situation, they found that the Cree activation, it turned out, was much more sensitive for detecting fluorescence from AV transduction than the direct fluorescence. And I would say that's also been our experience. So if you look at our own data on the right, I'm showing you AV9 transduction in 3 different tissues. On the top is liver, in the middle is gastrocnemius, and the bottom is adrenals. And if you look on the left side, you can see there's this gradient of ZS green fluorescence, right? You can have 1 copy of virus, 20 per cell, 100 per cell. Some cells are going to be really bright, some are going to be dim. And so that's basically what we see with this. In contrast, the tomato signal is a binary on or off. Once you've recombined that reporter allele, it's permanently on. You're always going to have a red cell. And so you can see right away that we have a lot more red cells here than we do green. So just in terms of answering the question, which serotype gets the most red cells in a given tissue, definitely the TD tomato Cree activatable reporter was definitely superior. And so I'm just going to show you some of the data, what it actually looks like. This is all available online. It's free for everyone to search and to look at for their own research questions. This is what AAV transduction looks like in the liver in terms of TD tomato fluorescence. You can see that at the dose we use, pretty much all AAV serotypes work very well, pretty much hitting all hepatocytes in the liver, with the exception of AAV3B and AAV4, where we have much more spotty fluorescence. Adrenals was kind of an interesting organ that we did not necessarily expect to see a lot of fluorescence going into the study, but we looked pretty closely at this and we see really good transduction of adrenals with most AV serotypes, with the exception of AV4 and AV3B once more. And we've done this across all 22 tissues that we looked at, and it's kind of interesting. If you quantify the fluorescence signal just by pixel counting, and you compare that to the biodistribution in terms of how many viral copy numbers are in a given tissue, there's this sort of sigmoidal relationship, right? So it tracks pretty good. At some point, once you get to liver, you're sort of maxed out. You can't get any more fluorescence, no matter how much more virus you add. We're already sort of saturated. But there's also tissues on the lower end where things don't exactly add up. So I'm giving you some examples here. So tissues like adrenals have a nice correlation, nice linear correlation between the biodistribution, in other words, how many copies of virus got in, and how much fluorescence we see. That's sort of true with liver, although liver is somewhat saturated already at this dose. But it's also interesting to note that some of the immune tissues, like lymph nodes, spleen, thymus, we didn't see much of a correlation at all. And that probably says that you're getting some AAV capsid taken in, but you're not necessarily getting expression in those tissues, which sort of makes sense for tissues that are rich in antigen-presenting cells, where their job really is to help you clean up infections. So one capsid I'm going to highlight here that came out of our work that I was particularly interested in was AAV4. So on the left, I'm showing you the AAV4 capsid crystal structure compared to AAV9, just with distance from center, color-coded. So you can see they look, at first glance, look very similar. In fact, they are. And if you align the VP1 subunit down below, you can see they are quite similar, with a lot of the differences being in these external loop regions here that I'm showing you on the bottom, where most of the differences are. So AAV4 was originally isolated from African green monkey kidney cells. It has this very unique affinity for alpha-2,3 O-link sialic acid. Interestingly, it does not use the AVR as its endocytic receptor. We don't actually know what the endocytic receptor is. Arvind Asakan's group has reported that AV4 shows a cardiopulmonary tropism, based on vector genomes as well as luciferase imaging. And there's not been much work of it clinically at all. So there's been a single clinical trial in the retina using AAV4, but beyond that, there's been almost nothing, which is kind of surprising. So this is a serotype that, as it turns out, maybe doesn't produce the greatest titers in the world, but has some very interesting properties. And we think that it's worth exploring further, and I'm going to show you why. One of the things we notice right away when we actually look at these tissues, this is the advantage of using these fluorescent reporters. You get information on the specific cell types that you're looking at transducing, not just what happens to the tissue as a whole. So what I'm showing you here is the retina, And we've labeled the vasculature with a fluorescent lectin just to show you where the endothelial cells are. That's the yellow signal. And then if we overlap that with a TD tomato fluorescence to show where we have AV transduction, you can see that there's excellent overlap between the tomato and the lectin signal, really confirming really strong transduction of endothelial cells in the retina. That was pretty impressive to us. In the liver, as I mentioned before, AV4 doesn't really do a great job. So looking at these livers that are also counterstained with lectin, we see, sparse labeling of hepatocytes with AV4, but nowhere near what you would expect to see with most other naturally occurring serotypes. But when we look at other tissues, this kind of pan-endothelial transduction pattern comes up over and over again. So this is looking at the brain. Once more, the lectin signal is in yellow, and we see really nice overlap of the yellow lectin signal with the red TD tomato signal, showing that we're hitting all of these capillaries in the brain really well with AV4 following a tail vein injection of the mice. In the lung lights up like an absolute Christmas tree. These sections are a little bit thick to do it justice, but I think it just makes the point that there's a lot of red tomato signal in the lung. Lung, of course, is a very vascularized tissue, and we'll come back to that in a couple slides. And then the stomach, I think, is also shows you a nice picture of what's happening here. There's a lot of vessels in the stomach, and a lot of these are also coincident with this tomato signal after we give AV4 delivery of Cre recombinase. Similar thing in adrenals as well, perhaps a couple other cell types transduced in the adrenals, but very good labeling of the vasculature overall with AV4 Cre. We see this across many tissues. Here is showing the thymus, and there's another, 8 or 9 others that you can look at in the supplements of our paper if you want to see if the vasculature in your favorite tissue is transduced well with AV4. And then last one I'm going to show you is the pancreas. We definitely see a lot of endothelial labeling in the pancreas that overlaps with the lectin staining in yellow. Interestingly, we also saw islets lighting up pretty well. And so that was exciting for us. Of course, islets are an important gene therapy target for type 2 diabetes, type 1 diabetes. And, we wanted to see what those look like. And so if we actually look at the staining in the islets, if we do an insulin stain for the beta cells, we can see pretty good overlap between insulin staining here shown in the green signal with the tomato signal for the AAV transduction. So we are definitely hitting beta cells in the pancreas, probably some other cell types as well. And so AAV4 has not really been thought of as a sort of a good cell type to go after islets with, but maybe it is. And then coming back to the lung, we wanted to take a closer look at the lung. As you remember, the lung was the single highest tissue hit with AAV4 in terms of vector genomes, per microgram of genomic DNA, but also the fluorescence was super bright. So what we did here was we repeated that experiment, except instead of doing tissue sections, we dissected the lungs, and then we digested them with collagenase, and then did flow cytometry to separate the endothelial cells from the non-endothelial cells. So the endothelial cells, in this case, are positive for PCAM, CD31 positive, and they're CD45 negative. And you can see here that in the right quadrant on the top right are our fluorescent endothelial cells. We see nothing in the negative control, and really good signal in the mice that received the AAV4. If we quantify this, we can see that both in male and female mice, we have in excess of 85% of endothelial cells in the lungs are transduced with AAV4. So really remarkable. We're starting to think about, what are some good applications for this. Some really nice work that was done, not by us, but by our collaborators at Jackson Labs. So this was Kathy Snow that did this work, was looking at the bone marrow as well as the blood for the hematopoietic stem cell compartment as well as immune cells. And so I'm just showing you here analysis from the bone marrow. These are heat maps of long-term HSCs, short-term HSCs, and various progenitor cells in male and female mice. And we were actually surprised to see pretty good transduction of several of these hematopoietic stem cells with multiple AAV serotypes, including AAV7, AAV RH8, RH10, RH74, in both sexes. And so that was really encouraging. This could be really important for, especially for genome editing therapies for like sickle cell disease, beta thalassemia, immune disorders, things like that. In terms of circulating immune cells, looking at peripheral blood leukocytes, we also see some signal here. CD11C positive monocytes seem to be transduced pretty well by a number of different serotypes. So that's also quite interesting. And so, hopefully I've piqued your interest in this data set. We're really happy and proud that we could try to put something together that's so comprehensive, where we're looking at 22 different tissues and also trying to provide some information about the particular cell types within a tissue that are transduced. And so all of this data is publicly available through the somatic cell genome editing toolkit on this website. If anyone would like to search it, you're welcome to look at all of our images and see what you can do with it there. And so to summarize this talk, surprisingly, there's only really been a handful of naturally occurring AAV serotypes that have been used for gene therapy. In many cases, we get something that works and we just stick with it. But I think there's a huge opportunity out there for exploring other naturally occurring serotypes that have unique tropism and unique properties that get us into cell types and tissues that are not particularly accessible right now. So the tropism of different AAV serotypes is important, not just for gene therapy, but also for basic science research applications for people that use mice to answer those questions. AAV4 we found has this very unique pan-endothelial tropism, although it largely avoids the liver, which we think is really a special property that we're going to follow up on. And also, I hope I've shown you the value of fluorescent imaging as a readout of AAV transduction, because it gives you this very, great sensitivity to see even rare transduction events within a tissue. And so with that, I really have to give a big thank you to the people that actually were, doing the hard work at the bench, in particular, Chris Walkie, who led these studies on our side. Chris is an assistant professor in the department who really led the biodistribution studies, as well as Kathy Snow at JAX, who really did the fluorescent imaging, the lion's share of the fluorescent imaging, as well as the hematopoietic stem cell work. And then, of course, Dan Wang was one of the main guys in Guangping Gao's lab, who actually designed and produced these very nice vectors for us to detect both direct fluorescence as well as Cree activation. And then with that, that's pretty much all I have. I hope everyone found it informative, and I'll jump on here in a minute from the pre-recording to answer any questions you may have. Thank you very much.
Thank you for that presentation, Bill. So let's move into some questions from the audience. So starting off, are cell culture experiments predictive of AAV delivery in vivo?
I think the short answer to that is no. They're not. They really aren't. In many cases, the receptors that are expressed in your immortalized cell line are not the same as what's in the tissue that you're working with. That's one major issue, but there's a lot of other factors that you really can't model well in cell culture. In particular, this issue of getting across the endothelial barrier is really a fundamental problem for accessing a lot of sort of non-hepatic tissues that is not very easily modeled in cell culture. So I would just really tell people to exercise caution in throwing some AAV on cells in a dish and assuming that's going to directly translate to their favorite tissue in a mouse. It usually doesn't.
And moving forward with mouse experiments, how are they translating to humans? How well is the data?
I think overall, I think some of the serotypes that work well in mice also work well in humans, at least for a couple different tissues, right? For liver, for skeletal muscle. There's been a fairly good translation that you can still deliver cargo. Are those the best serotypes? Is the best serotype in mouse always the best serotype in humans? In many cases, it's not, and we've seen a lot of examples where that's not really borne out. For example, like AAV3 and derivatives of that are very good for human liver, but not really in mouse liver. So there's always exceptions, but I would say by and large, something that works in a mouse will also work in a human. It's just a question of how well.
Sure. And why are there so few naturally occurring AAV serotypes been used for human gene therapies?
Yeah, that's a great question. And I don't think it's really a question that's driven necessarily by the science. I think it's been more of a practical consideration that, companies and investors are going to stick with things that are proven and safe. And so in many cases, just piggybacking on what's already known or what's already been tried before in order to get through the regulatory process and really minimize risk. But unfortunately, that approach may not really be the best. And you're really missing opportunities to have more effective gene therapies by investing more heavily earlier on in terms of really finding what is the best serotype for your tissue and your cell type.
And what are the challenges with engineering the AAV capsid to target new tissues or cell types?
Yeah, I think there's a lot of challenges. I think one of the biggest challenges is this sort of three-body problem that there's a lot of things we just can't do in humans, right? So we have to choose an animal model that recapitulates the human situation as best we can. But even if you go to a non-human primate, they're still not humans. And so I think there's fundamental species differences, especially in terms of what proteins are expressed, how those proteins are decorated with the different carbohydrate groups. That varies a lot. And that's really a hard thing to kind of sort out. And I just think there's a lot that really is not easily modeled, unfortunately, in smaller animal models. So you really need kind of multiple approaches to address these questions.
Fantastic. And could you explain the difference between the genders and AAV tropism? I think just briefly touching on maybe gender differences and how they affect tropism.
Yeah, so I would say sex differences, just to be precise. And I think one of the big things we know, at least in mice, is that in the liver in particular, there's a very strong bias in terms of better transduction in male mice than in female mice. And we don't really know all of the reasons for that. There's been a little bit of work on it. There probably hasn't been enough. But by and large, male mice will transduce better in the liver than females. In some cases, some serotypes, you might get kind of the opposite effect, where kind of peripheral tissues in the females might be slightly better transduced than the males. And we don't really have a good answer for why that is. Some of it could be receptor expression, but a lot of it probably has to do with downstream effects on, either formation of episomes, stability of episomes, or transcription from the episomal genome. Yeah.
And kind of following up on that, it looks like someone wants to know if in clinical trials, is there plans to stratify male versus female?
Yeah, in general, there's not. And one of the problems is that for you're often dealing with a very rare patient population, right? And so you have to basically take what you can get. And even starting with the very rare patient population, not everyone is eligible to receive the gene therapy. They might have to meet certain conditions in terms of the status of their disease. But also, we also have this issue of, pre-existing neutralizing antibodies, which is also a major problem that might exclude half of your patient population. So the numbers of people that are eligible to go into a clinical trial are typically very, very small, and it's often not, restricted, or they're not thinking about it that way. And unfortunately, a lot of the best, most effective ones, at least for liver, are X-linked diseases, right? So the hemophilias. And so we don't have any information really on females in those particular situations. So I do think it's something that needs to be explored further, and we should think about what the best way to answer those questions are from clinical trials where both males and females are being treated, and what are kind of the sort of longitudinal readouts we can get of transduction that might be separate from the kind of conventional readouts of disease correction.
Perfect. I do want to remind the audience that to stay tuned for the last presentation, which will talk a little bit more about sex-based differences. So if you're interested, please stay tuned. And the final question, which AAV has preference for microglia?
That's a good question. I'm not sure there is an AAV that has a really strong preference for microglia. I think a lot of that has to do also with your route of administration. If you're doing a direct injection into brain or intrathecal injection, a lot of different serotypes can hit a number of different cell types within the brain. If you want to go the kind of systemic route with an intravenous delivery, you have fewer options. Although there's some really nice capsids that have recently been developed, like Voyager has a really nice one that gets across the blood-brain barrier in mice and primates now. There's other ones out there as well. But I actually don't know the current state of knowledge in terms of what is the best capsid serotype for microglia. I'd have to defer to people that are more in the weeds in terms of CNS delivery of AAV.
Fair enough. Well, thank you so much, Bill.
All right, thank you.
All right, so let me introduce our next speaker. Ajinkya Sase is an application scientist at Vector Biolabs with over 20 years of experience in molecular and cellular biology. He earned his PhD at the Medical University of Vienna, where he developed new drugs targeting the dopamine receptor to improve learning and memory. Before joining Vector Biolabs, Sase was a senior scientist in the gene therapy industry, leading AAV vector engineering from construct design to building pre-investigational new drug studies. He also held research roles at the University of Pennsylvania and Medical University of Vienna. Known for his collaborative approach and problem solving mindset, Sase combines hands on vector engineering expertise with a passion for helping researchers advance their gene therapy goals, whether optimizing small preclinical studies or scaling towards clinical development. He is the primary scientific resource for Vector Biolabs, customers providing expert guidance on vector design, serotype selection, and project strategy. Let's make sure your slides are up and running. Okay, take it away.
Hello, everyone, and welcome to my presentation. My name is Ajinkya Sase, and I'm an application scientist here at Vector Biolabs. I'm very excited to show you how customized viral vectors can help you solve your research questions and then set you on a path to the success. Here's a brief agenda for my presentation. In this presentation, I will discuss important aspects of viral vector design that can help with increased tissue targeting as well as specificity. Then I will show you some real-world example of how dual AAVs can be used to successfully deliver large genes. And at the end, towards the end of this presentation, I will show you how control viruses can be resourcefully used to gather some preliminary data that you can use to set up your research on path to success. Before this, let me talk or introduce Vector Biolabs to all of you. Vector Biolabs was founded in 2004 on a bench in University of Pennsylvania. From then onwards, it expanded into a 20,000 square feet production facility just outside of Philadelphia in Malvern, Pennsylvania. We are experts in viral vectors production, both AAV as well as adenovirus. We are industry leaders in end-to-end production. End-to-end means we will help you design the construct, we'll help you, our cloning team will clone it, and our packaging team will package it in your choice of viral vector. We have a highly experienced team. Most of our team members have a tenure of more than 10 years working with Vector Biolabs. Over these 20 years, Vector Biolabs has gathered more than 5,000 citations. Currently, Vector Biolabs offers AAV as well as adenovirus viral vectors. Since we are talking about viral vectors, it is important to choose the right vector platform that is best suited to answer your research goals. And here's a simple schematic of these two viral vectors that describes briefly what is the packaging capacity, how they work. The maximum packaging capacity of an AAV is 4.7 kilo base pairs. For some of the engineered capsid, it can increase a little bit further, but it's close to 4.7 kilo base pairs. But in case of adenovirus, the maximum capacity for our adenovirus vector is around 8.5 kilo base pairs, so almost double of that AAV. On the next slide, I'll show you some of more detailed differences between the AAV as well as adenovirus, so that when we are selecting a viral vector to address your research questions, you can consider these options. As many of you know, AAV comes in two formats, single-stranded AAV as well as self-complementary AAV. And they both provide some pros and cons compared to adenoviruses. In this table, you can see the cargo capacity for single-stranded AAV is 4.7 kilobase pairs, whereas for cell-complementary AAV, it's 2.5 kilobase pairs. It's almost half. The advantage of using self-complementary AAV is that the onset of expression is quicker than single-stranded AAV, but you have to deal with a smaller cargo space. Similarly, adenoviruses also have a quick onset of expression. Within 24 hours of transduction, you will see onset of expression. Adenovirus provide the possibility to deliver larger genes up to 8-9 kilobase pairs, but they come with a caveat that adenoviruses are highly immunogenic. That means you will see a lot of immunogenic response depending on route of administration as well as tissue. So this can lead to various complications in your study. Considering these various pros and cons of single-stranded AAV, self-complementary AAV, and adenoviruses, you can strategically select or plan a suitable viral vector for your study. A next consideration in achieving cell-specific targeting is once you have chosen the viral vector for your study, answering following questions will help you in achieving this cell-specific targeting to improve targeting. The first is the type of study. Whether your study is an in vitro study, it is in vivo study, or are you going to combine both, start with an in vitro study and then move on in vivo study with mice, rats, or NHPs? What would be the route of administration? Are you going to deliver the viral vector via tailwind, or is it going to be intrathecal? Is it going to be intracerebroventricular or through intraorbital veins? So these considerations will help you deciding in what kind of viral vector platform and what kind of AAV serotype, what kind of promoters you can use to achieve this targeted cell-specific expression of your gene of interest. Some of the examples of these achieving cell-specific targeting, and I'm giving here a quick example of transducing microglia. You can see there are multiple AAV serotypes that work really efficient in vitro setup. For example, AAV2, AAV5, AAV6, AAV8, and AAV9. They work really well in vitro setup, and you can achieve up to 80% of transduction. Some of these serotypes, for example, AAV6 or AAV8, will show up to 80 to 30, 40% transduction. And you can see here, there's a significant difference for each naturally occurring serotype in the transduction efficiency. When you move or when you consider in vivo delivery of these viral vectors, some of the viral vectors work really and efficiently well when they're directly injected into brain. Whereas some of the naturally occurring serotypes, AAV6 or AAV8, when delivered intravitreally or subretinal, they show better transduction as well as a simple route of administration, a non-invasive route of administration instead of stereotactic or direct parenchymal injections. But in some cases or some research questions, one definitely wants to use a direct parenchymal, very localized injection. In that case, AAV9, which is used widely, shows very efficient gene delivery. Combining this naturally occurring serotypes with specific ubiquitous promoters or cell type specific promoters, for example here IBA1, can help you in achieving this precision and efficient transducing of microglia. On the next slide, I'm showing you some of the engineered AAV capsids that Vector Biolabs can provide you that show improved cell targeting and specific to neurological research purposes here. Each of these capsid has its own benefit depending on your research question, depending on type of cells, number of cells you want to target. And at Vector Biolabs, we can help you in deciding more appropriate capsid based on research goals. Now, some of these capsids target many different cells in nervous systems. They show, some of these capsids show distributed cell type specificity. They might transduce certain portion of neurons as well as certain portion of non-neuronal cells. So that's when we move to a next stage of viral vector design. And that is using cell type specific promoters to restrain your gene of interest expression in specific cell types. On the left side here, I have shown a list of neuron-specific promoters. HCN is a very common promoter that expresses in most of the neurons, whereas VGAT promoter restricts the expression into GABAergic neurons. One can consider targeting dopaminergic neurons using tyrosine hydroxylase promoter so that it's restricted into dopaminergic neurons. On the right-hand side is a short list of non-neuronal promoters. One of the most common non-neuronal promoter is GFAP. It's astrocyte-specific promoter. There are a few different iterations of this promoter, and depending on your interest, we can select or choose specific GFAP promoter. There are microglia-specific promoters. MBP is another specific promoter for targeting oligodendrocytes. If you are interested in targeting Purkinje cells, what we call is L7-6 is another Purkinje cell specific promoter. And I encourage you to visit our website for a detailed list of various promoters, both neuronal as well as non-neuronal promoters, to take a look at that. Depending on the cargo that one wants to deliver using these viral vectors, you can choose between a shorter promoter or a longer promoter, along with any reporters like GFP, mCherry, or if your system involves a Cre-lock system, a lock system of gene delivery can be also achieved with these cell type specific promoters. Another example of achieving targeted cell type specific expression is that we can synthesize a promoter of your choice or selection based on if you have any preliminary data. For example, if you have some sequencing data that shows grouped cells in particular cell type, and you know the biomarker or some marker from these cells that separates these cells, we can help you synthesize that promoter so that the perturbation of those cells can be achieved with that specific promoter as well. On this next slide, I'm showing you some of the previously published literature that summarizes how in combination of engineered AAV capsid and different promoters, either ubiquitous or cell type specific, can affect transgene expression, either in vitro or in vivo setting. Here you can see these are some of the modified capsids. This is a self-complementary AAV6 with a cell type specific promoter F4/80 that shows up to 95% transgene expression in vitro setup. Whereas when you move with this combination, it can go down a little bit towards 70-75% when delivered directly into the brain or directly into hippocampus of newborn mice. Whereas the ubiquitous promoters like CMV, which is very common, can lead up to 80% transgene expression in vitro setup. Whereas with an cell type specific promoter, even though it is cell type specific, you will see some level of off-target expression because that promoter would be expressed or active in neurons as well as some non-neuronal cells. For example, CD11B. But depending on your research question and how many or which type of cells you want to target, you can always choose a combination of capsid and promoter to improve specificity and efficacy. So scientists have been using this approach of engineered capsids and selective promoters for targeting various genes, targeting various cells for a very long time. There has been a lot of literature. There's a lot of information available on the internet. At Vector Biolabs, we can help you in choosing right capsid for your question and right combination of promoter. We have many engineered capsids as well as promoters available. And I again encourage you to look at our website linked on these pages, where you can see the number of capsid, the number of promoters. And if you still have any questions, you can always reach out to me. The next question that is critical for delivering AAV or gene of interest in the research. AAVs are being used in research for a very long time. The applications include from gene therapy to disease modeling to solving complex biological questions. But a critical limitation for using AAVs is delivering larger cargo or delivering larger payload or larger genes which are larger than 4.7 kilo base pairs. As you can recall from previous slide, the maximum packaging capacity of AAV is 4.7 kilo base pairs. And this limits its use for delivering larger cargos. So how do we overcome this cargo limitation? Many research groups all over the world have been trying to solve this issue with dual AAV as well as triple AAVs. And I'll walk you through some of different options that are available for delivering cargo that is larger than 4.7 kilo base pairs. One approach is it's called transplicing dual AAV. The second approach is overlapping homologous recombination. And the third approach uses the intin hybrid dual AAV. And after that discussion, I'll show you how Vector Biolabs produced a dual AAV system that was successfully delivered in animals and shows robust behavioral phenotypes. Here is the first example of transplicing AAVs. It has been used widely to deliver dystrophin. There is large literature volume available. You can look for it. At the same time, let me give you brief information about transplicing AAVs. In this system, the GOI is split across two vectors so that it can be packaged in two different AAVs. The first half, or towards the 5' end of the GOI, is fused with a splice donor, called as a splice donor, and the 3' half end, or 3' end, has splice acceptor. During the transcription and translation, this splice donor and splice acceptor is removed by splicing, resulting into a full-length microRNA. So you deliver both the AAVs containing the splice donor and splice acceptor sites via your choice of route of administration, either intravenous, either intrathecal, or ICV, or intranasal, as you can see from few next slides. The transduction takes place, and any cell that receives both the viral vectors will show this intron splicing to produce full-length mRNA, then which is translated into a full functional protein. This requires co-transduction of both AAVs. That means you need two AAVs and efficient splicing system with the splice donors and splice acceptor sites. A second approach is overlapping homology, where each vector contains an overlapping gene segment, which can range from 300 to 1,000 base pairs depending on your gene of interest. And this leverages natural biological system where homologous recombination can take place, resulting into a full-length cDNA. And this full-length cDNA then can be transcribed into a single mRNA and a full functional protein. A third approach is based on the third biological system of protein splicing. This has been used to deliver ABCA4 gene, where the two AAV vectors contain a protein splicing junction called an intine, where these intines act as, again, a very similar way of RNA splicing, but at a protein level. These protein domains that mediate self-excision from each other will lead into a functional protein, sliced protein after transplicing. So the N-terminal of intine and carboxy terminal of intine is removed, and N-terminus of the half the protein is fused with the C-terminus of the remaining half protein, resulting into efficient splicing and functional protein. So these are three different approaches that have been used, that are under development, and many have published on for delivering larger cargo using AAVs, using dual AAVs or triple AAVs as well. So in some of the next couple of slides, I will show you a case study where Vector Biolabs developed dual AAV for delivering a larger cargo to neuronal cells. Here, Vector Biolabs designed and engineered and produced a dual AAV9 vector system to deliver CRISPR-based system that can then improve or help alleviate symptoms of anxiety and memory function. The customer chose AAV9 because AAV9 crosses blood-brain barrier to a certain level. The route of administration was intranasal delivery. Why dual AAV? Why they use dual AAV? They wanted to deliver CRISPR-Cas9 system, and we know the Cas9s are really big. They're super big. They can go up to 6-7 kilo base pairs. So Vector Biolabs split the SP-Cas9 into two vectors. One is with the N-terminus, and the remaining was split with a guide RNA construct and a spliced acceptor site. So AAV9M is CP2 promoter with SP-Cas9, which is around 4.7 kilo base pairs, and then another AAV expressing the selected guide RNA under a U6 promoter and a GFP reporter. So that when co-transfected or co-delivered, these two AAV9 will result into specific knockdown of HTR only in cells that receive both the AAVs. So Vector Biolabs can help you design these larger genes that can split across two AAVs successfully. And using this technology, this dual AAV, this group of researchers showed that successful delivery of two split genes can result into reduced neuronal excitability, This then further translate into reduction in HTR mRNA expression as well as reduction in protein that leads to improved behavior. Improved behavior in terms of reduced anxiety and improved memory performance in these animals. This company patented the guide RNA sequence used in this work, but this system can be modified and used with other guide RNAs, or we can help you in using this system for other transgene delivery, not just CRISPR, but if a larger gene of interest you want to deliver to these cells. Now going towards control vectors, towards part where you can use how this dual AAV with a GFP control can help you, and how vector biolabs can help you on the path to research success. The control vectors are very important. They can help you understand how the viral vector is going to function in your cell system. You can determine some of the preliminary information from use of control vectors, like vector dose, what would be the level of expression, are you going to target the desired cells, number of cells, and the level of expression in those cells. It can help you validate the route of administration, and it will provide you this preliminary data and give you a chance to establish your system so that you have more confidence and use this data in your grants as well as funding applications. This will save a lot of time in pre-hand and help you in a long-term run, because then you have a lot of variations. Many of these control vectors we have them off the shelf. That means they are ready to ship. Again, they come with many different reporters like GFP, mCherry, TD Tomato. If you have any other specific reporter or you want to analyze or assess a specific promoter, we can help you produce these control vectors with those specific promoters so that you can analyze before you invest large amount of money. And we know that in current funding scenario, everybody wants to use their money in a very distinct ways. So given that, these control vectors can help you with achieving this data, preliminary data, and assessing your choice of promoter, choice of capsid for your research study. On the next slide, I'm showing you some in vitro data that we generated for this dual GFP control with plasmids. These are bright field images here. You can see all three pictures here show efficient transduction. The cells are survived the transfection. And on the next slide, you will see that only the cells that receive both the plasmids show GFP expression. So not one AAV, not a single AAV, but when you deliver a combination of plasmid, AAV plasmids, the dual AAV system works and shows GFP expression. Now this is with a very ubiquitous promoter called CMV, and we can help you design this or replace this CMV promoter with specific promoters of your choice so that you can test these plasmids. You can test these control plasmids in your cell system to assess the expression levels. You can conduct some preliminary studies to find out the MOI that can be used, whether your choice of promoter works in this cell or not, what is the level of expression, and how you can use this information for your in vivo studies. This is another slide showing single plasmid transfection with a dual AAV plasmid transfection. And I wanted to show you this because I get this question often, how well this is different than single. So you can see that even though there is much difference in the expression level, you have now much more, or more than 0 cells that can express your gene of interest. I would be happy to take any questions on this after the talk as well. So Vector Biolabs is your research partner. We have end-to-end production. That means we help you design. We have a dedicated cloning team. We have a dedicated production team, and we have quality controls at every step. One of the most important point, and I feel very proud about it, is that we offer fastest turnaround time in this industry. For if you have a packaging ready AAV plasmid or adenovirus plasmid, the packaging takes between 2 to 3 weeks. If there are any custom steps that requires cloning, usually it's between 4 to 5 weeks. We have expert design that supports. We have more than 100,000 pre-synthesized gene library that you can choose from. And if you don't find it, let me know. I can look through our database. Another option is always that we can synthesize your gene of interest and then use it for cloning. We have a capacity of running more than 100 plus parallel preparations. We have an optimized production process that way we can produce many more different viral vectors at the same time, so that you have all the different vectors, your customized vector, your control vectors together, so you to start your study. We're 100% manufactured in USA in one facility, just as I mentioned, outside of Philadelphia in Malvern, Pennsylvania. Average tenure of our team is between 10 to 20 years. We just celebrated one of our production team members' 20 years anniversary. Over these 20 years, we have gathered more than 500 citations, and we can always support you with a large quantity preparation to support your non-human primate studies as well as large animal studies for research purposes. At Vector Biolabs, quality is always an important consideration for us. We offer as our quality, we offer whole plasmid sequencing. That means the final plasmid that is used for packaging in viral vectors, you have that sequence available. We analyze the purity of the final viral vector preparations using SDS page, a very standard, and you can appreciate from this, our viral vector preparations are more than 95% pure. We analyze some of the aggregation as well as full and empty capsid ratios using stunner machine. We perform titration with standard qPCR method. We also offer titration by dPCR using QIAGEN's QAcuity system. If you are interested in analyzing the actual morphology of the viral vectors, we can provide you TEMS analysis, transmission electron microscopy analysis with our third-party collaboration. And we also can carry out endotoxin preparation for these final viral vectors. And I'm very happy to say that our endotoxin levels are much lower than that is required by regulatory agencies. And with that, I'll end my presentation. Thank you so much for your attention. I'll be happy to take any questions.
All right, thank you, Ajinkya. Let's take a few questions from the audience. All right, let's start off with how quick and slow is expression of SSAV versus SCAV?
Can you hear me? Can you see me?
Yep, I can hear you fine.
Yes, that's a very good question to start with. Both these options of single-stranded AAV and the self-complementary AAV offer options depending on their research question. Self-complementary AAVs often show expression within 24 to 48 hours of the dosing, whereas single-stranded AAVs can take longer than that because how both of these AAVs are um, processed in the cell. Because of that, self-complementary AAVs show expression within 24 to 48 hours.
And how should someone choose between the two?
That's another good question. The main difference between these two is the packaging capacity. The self-complementary AAVs have a packaging capacity of half than the single-stranded, so around 2.5 kilobase pairs. So that means if your gene of interest or the cargo is under that um, payload size, you can choose. And if your research question requires immediate transgene expression, then the choice is to go with the self-complementary. But if the cargo is larger than that, which is most often the case, and you want a long-lasting expression, in that case, one need to choose single-stranded AAV with around 4.7 kilobase per size.
Perfect. And what kind of quality controls are necessary for if I was to test a vector, or is there some kind of quality control analysis that I need to perform to make sure that it's up to standard?
So the most common quality control, as I mentioned in the presentation, is looking at the plasmid that is used in production of the AAV, and that's why we provide the whole plasmid sequence of to the customer so they can analyze and look for any there are no unintended changes or mutations in the gene or the payload that can hamper your research questions. And we ensure that by providing whole plasmid sequence that was used in packaging or production of the AAV. And the second is the purity of the AAV. We want you want a uniform, purified AAV that can that has maximum packaged your payload so that you are delivering right amount of your transgene to these cells. Another important quality control is the endotoxin levels, because they can elicit immunogenic reactions in animals. So when you are considering that, we provide a very low endotoxin uh, levels in our AAVs. So these are some of the major quality control or steps that one should consider when using viral vectors.
And if someone was going to get your company to actually make the vector for them, are these the same kind of quality control that you would perform and then provide analysis, like a report of?
Exactly. Yes. So these quality control analysis is included in our production process, and these are provided with your vectors.
Excellent. And how long does it take for you to make a custom virus?
Most commonly for a simple one-step or two-step clonings, we have a dedicated cloning team, and we can custom produce the viral vector right from the beginning of cloning to packaging and purified AAV in 4 to 5 weeks. If you have a ready plasmid that can be packaged in viral vectors, we can package it in 2 to 3 weeks timeline.
Excellent. And if you have a GOI plasmid, but it's not an AAV plasmid, can you still use it?
Yes, I get this question also very often. We work with different, we can work with many different type of starting materials. If your GOI is in a non-AAV plasmid, we can we can still use it for cloning. We will clone it into our AAV plasmid backbone and then package it. Sometimes for smaller fragments, you can send us gene fragments that we can use as well for cloning. So your GOI can come from different plasmids, no problem. It can come in fragments, and then we can clone it here on site in our AAV backbone plasmids.
And how do you determine the right promoter and capsid combination for a specific tissue or cell type?
A combination of specific serotype and promoter requires some experimental work with few different selection of serotypes and testing them, as I mentioned, with some control vectors like that shows that you can choose either with the GFP reporter or mCherry reporter. So selection of right capsid and promoter requires preliminary experimental work using many different available control vectors that we have.
Fantastic. And what are the current limitations of using microRNA or tissue-specific promoters and AAV vectors for targeted transgene expression?
MicroRNAs are very interesting tool that can help you detarget or help you limit the expression in off-target tissues. Specifically, there are a few microRNAs that are widely used to detarget liver expression of the transgene. But along with that, microRNAs can also limit the expression of your transgene in other tissues. So combining sufficient or limiting the number of microRNAs in your construct will help in answering this question.
Perfect. Well, I think we have one more question. How do different AAV serotypes compare in terms of crossing the blood-brain barrier and transducing cell types like microglia, astrocytes, or neurons?
Yeah, so most naturally occurring AAV serotypes have a limited blood-brain crossing capacity. Most commonly used AAV9 can cross, but engineered capsids like PHPEB that we heard in the very first talk can very efficiently cross blood-brain barrier after intravenous delivery. But that again limits that PHPEB can work in some mouse strains, but not in all mouse strains. There are a few other engineered capsids that are more efficient in crossing blood-brain barrier. There are other engineered capsids like AAV, MG that can cross blood-brain barrier as well as target microglia. Then there are AAV5 is more efficient in transducing astrocytes than other AAVs. So based on your research questions, you can choose between AAV9, AAV5, or AAV-BI3, AAV-RH10, and which cell you want to target. But I would also like to add, it's important to combine that with a specific promoter so that you have more controlled expression in your cell, specific cells.
Great. Well, thank you so much for your presentation, Ajinkya. And if anyone has any further questions, please reach out to the speaker. He has his email on the screen. All right, so let's introduce our next speaker, Zhongming Zhu. Zhu is a Chancellor's Professor and Director of the Center for Neural Circuit Mapping at the University of California, Irvine School of Medicine. His research interests include cortical plasticity, learning and memory, brain disorders, and the development of advanced viral genetic technologies for studying neural circuits. Zhu's work holds significant translational potential, aiming to advance treatments for neurodevelopmental and neurodegenerative diseases. Let's make sure your slides are up and running. Okay, Zhongwen, take it away.
Okay, hello. My name is Jiangming Zhu. I'm a Chancellor's Professor and Director of the Center for Neurocircuit Mapping at the University of California, Irvine. I'm very pleased to participate in the Scientists Cell and Gene Therapy Symposium. For today's talk, I will first tell you about our Center for Neurocircuit Mapping, its infrastructure and resources related to the new viral genetic tool development. And then I will tell you about our recent work to develop enhanced AAV to target specific brain cell types for therapeutic applications. The UCL Center for Neurocircuit Mapping focuses on neurocircuit studies and new neuroscience technology development. We are particularly strong in developing new viral genetic tools for neurocircuit studies. And we're interested in understanding and defining molecular and neural mechanisms underlying brain disorders, including as Alzheimer's disease. So as you can tell from here, this center actually emphasizes the collaborative inclusions of physical sciences, engineering, and computer science faculties on UCI campus and beyond UCI, because as we all know that a neuroscience field is increasingly becoming multidisciplinary. So our center members, we actually have a pretty large center now. We have about over 100 faculty members from both inside UCI and outside UCIs. And center members work collaboratively to establish transformative and translational research programs and focus on our ultimate goal to develop cures for treating human disease of the nervous system. The center members are connected by shared grants, collaborations, and we have both internal members and external members. And many of our members are funded by Brain Initiative and other major consortium organizations. And at our center, we have 3 research areas of focus, including neurocircuit mapping in healthy disease. As I mentioned, we do have a major emphasis on Alzheimer's disease research. And the research areas of focus are also including viral genetic tool development. This is highly relevant to today's talk. And part of this program is turning into a gene therapy and translational research of genetic disease. And lastly, we do have recently, we do have new directions in single cell technology development, particularly in single cell and spatial multiomics and related therapeutic discovery. At our center, I'm quite proud to say we have established ourselves as a critical hub for cutting-edge technology and resource development in biomedical research, not only within UCI, but also extend beyond the UCI. And this is facilitated through the dissemination of our resource alongside education and research initiatives, such as seminar, symposium, and conferences. And our annual center conference now has become well known in the field. For some of you, probably might be interested to come to our center events. For example, this year we just had a very successful 2025 conferences. So leveraging the federal funding and the UCI school medicine support, we have established the critical infrastructure and resources at our center. And so these resources, particularly, I would like to highlight our viral production facility. The center maintains a robust, scalable infrastructure for viral vector production and distribution. And shown up in these slides, our online, our center supports requisitions from both UCI and external investigators. We have supplied materials to more than 125 laboratories across 67 US domestic institutions as well as international institutions. And our centers also participate in the BRAIN Initiative Armamentarium Consortium Viral Resource Network. And I should say that we're open for collaborations, and where we would like to contribute more to the tool development toward the gene therapy applications. So our production team support investigators worldwide by developing and producing high-quality viral vectors for research applications. We actively develop a range of viral vectors, and we're strong in developing new tropical viral vectors, including herpes, rabies, yellow fevers for new circuits mapping or new circuits tracing studies. And part of our center or the team is supported by the BRAIN Initiative Armamentarium Consortiums. So the Armamentarium project, the funding supports our team to distribute the resources, including recently developed enhanced AAVs reagents to abroad neuroscience communities. And so if you go to our website, center website, you will see our viral vectors catalog page, including enhanced AAVs. And that is, actually do have a good number of the viral reagent to offer, and it's easy to use. And if you're interested in some new development viral tools, you can go there to check out, and you can also place order and contact our customer service. So with this introduction, and I'm going to turn into today's talk about enhanced AAV development and applications. So you may wonder, what are enhanced AAVs? Enhanced AAVs are new generations AAV vector that incorporate DNA enhanced elements to improve the cell type specificity, efficiency, or regional targeting of gene expression of AAVs. And they're also called enhanced-driven AAVs or enhanced AAV vectors. So like conventional AAVs, there's engineering the viral vectors to use to deliver genetic materials. So different from conventional AAVs, sometimes those AAVs use promoters and to drive the targeted gene expression. So instead they use promoters, enhanced AAVs use the enhanced element, essentially CREs, the cis regulatory element to drive the targeted gene expression. And I should mention that if you look at the enhancer, there's at least an order of magnitude more of the CREs. And these are the distant, CREs are distal genomic regions that help to regulate well and when promote the transcribed DNAs. But this number of the CREs are order of magnitude more than the promoters, and which suggests to me that enhancers may be more cell type specific. So that's one of the rationales for us to develop enhanced AAV to target specific cell type and drive the cell type specific gene expression. And once you identify those enhancers, you can insert this enhancer upstream as a minimal promoter in AAV backbone, and we actually will be able to achieve the gene expression patterns in specific cell type. And so these enhanced AAVs, I should say, is relatively new development, as I'm going to tell you in the next few slides. So related to the enhanced AAV tool development, I think you may be interested to check out the NIH news release on May 21st of 2025. And then so Unistri's basically tell us about the scientist designs, the gene delivery systems for cells in the brain and the spinal cord. And they state that NIH found, these are a group of studies are funded by NIH, particularly by Brain Initiative Armamentarium Consortiums. And these NIH funded studies, the basic technology breakthrough could enable targeted therapy for many neurological disorders. So here I'm showing one of the images from taken from the Allen Institute's the news release page. So you can see that different population of cells in mouse brain. Each one now basically each one a color code. Each cell type is color code, and each one of them targeted with high specificities by one of the many enhanced AAV AAV reagents developed by the armamentarium investigators. You can see different, some are quite restricted, so some are just really targeted striatum, in particular there's MSA and those GABA neurons. And so clear you can see that some there's some cell type is more broad, some are more region specific. So those tools, those new tools now really I do think it's a promising tools, allow us to a more precise targeting cell type or different regions in the brain. And the enhanced AAV platform, I believe they will have the potentials to transform how scientists can study neural circuits. And it provides researchers with gene delivery systems for various species used in research. So without the need for genetic modified or transgenic animals. This is quite important. And some studies published in the collection of studies, published in collections, the articles I'm going to show later. So including to visualize the fine structures of brain cells with fluorescent proteins, and also some studies they can use those enhancers to target specific cell types to activate or silent circuits, control behavior and cognition. And also very important, I should mention that since AAV-based treatment is already approved for some conditions such as SMA. And the newly collections of tools, gene deliver, particularly enhanced deliver resources, and lays the groundwork for more precise targeting or more precise treatment that can only target the effector cells in brain, spinal cord, or brain blood vessel. So this, I do think the recent technology does have a huge significance and impact. So this is so the collection of the paper the collection paper was published along with the news release on May 21st, 2025, and these are nice, very nice studies that describe a range of methods to gain access to neuron or non-neuron or cell types within the central nervous system. And particularly, I should say that it's the enhanced AAV tools are highly highlighted in this collection of studies. And I should say that our center investigators contributed to the 8 papers published in the collections. And today I'm going to focus on one of the papers and one of the studies related to specific targeting of brain endothelial cells using enhanced AAV vectors. And these studies was led by Dr. Eric Velezig River in my lab at the Center for Neurocircuit Mapping. So specific targeting of the endothelial cells that underline the brain, blood-brain barriers functions has been was a long-standing change. So we know that the BBB is specialized, centralized, the vascular system, that confirm highly privileged the chemical and cellular access to the brains relative to the circulation in the rest of the body. And damage and the dysfunction of the BBB is associated with age-related changes in cognitive function and neurological disorders, including Alzheimer's disease and traumatic brain injury, among others. So the brain endothelial cells, BEC, they are important because they are the only neurovascular unit cell type that are topologically continuous across the highly heterogeneous BBB. So one strategy is target the BBB is to focus on BECs. So in the past, and this is I'm sure say that the field, the investigators has been certain success in targeting the BECs using the AAV capsid engineering. And these capsids has been previous genetic engineering to confer the specificities to endothelial cells. However, these tools or these capsids, do show limited endothelial cell specificity that varies by delivery conditions. Anyhow, they still have significant limitations. And shown up here, I summarize some early works. The AAV-BR capsid, this capsid was first reported EC selective capsid, and turned out actually is depend on. I would say that this BBR did some good job, did some jobs in targeting endothelial cells, but still there's a lot of leak of labeling the neuronal cells in the brain. And a recent development, including V1 and BRI30 and AAVX1. AAVX1 actually among the capsid seems to probably work better than others. But still, there's some different issues as listed here. So consider the recent the technology development to enhance AAV. So we decided for our work, we would decided to take a different approaches, leveraging the driven gene expression technology. So as shown up here, illustrated here, so recent AAV vectors have incorporated gene regulatory elements such as enhancers to target specific type of neurons and other brain cell types. And so you may ask me, how does the technology work? Right. So this as listed here, this is in terms of the technology or conceptual design is relatively straightforward. First, we need to identify the enhancer elements, right? And so there's a broad database, a lot of resources, including we can look at basically look at chromatin accessibility assays, including ATAC-C, ChIP-C, as well as DNA methylations. And so we'll be able to identify the regulatory sequences, right, in certain cell types. And so there has been some pioneering work, done. So it really is about 5 years ago, right? So as shown by one of the Allen Institute, it's earlier work, right? So you identify the enhancer, cell type specific enhancer based upon single cell ATAC-C data. And then you pick up those a few hundred BP of these DNA elements and clone into the AAV backbones. Paired with a minimum promoter to drive the report expression or effector gene infection. And then after you make the virus, you can the rest of the step is basically just to characterize and test and to confirm if these enhanced AAV work and how well it work. Right? And oftentimes, after the careful computational analysis, you pick up the top candidates. Oftentimes, the enhanced AAV work well. Okay, so now with this, I'm going to get into tell you about the details of our work. And this slide is showing the schematic workflow for the design of the BEC enhanced AAV vectors in our work. Bing Lin who used their uses and our group has substantial collaborations. So we leverage the resources in the catalyst as a basic cis element atlas that Bing Lin's lab developed over the years. And from the 1 million candidate enhancers identified in Catalyst mouse atlas, and there are about, there's more than 2,700 candidate enhancer sequences identified in the industrial clusters, in bone industrial clusters. But only 83 of them identified exclusively in the industrial type clusters. And one of the top candidates who identifies CRE 89763 is among of the 83s. And so we pick up these top candidates along with a few other enhancers. And turn out the CRE 8973 work very well. So once I identify these enhancer element, so we package, we clone this into the AAV backbone, and we package into the AAV virus using the AAV PHEB capsid. So AAV PHEB capsid is facilitate the viral entries across the BBB. So it allows we use this quite often to do the whole brain-wide transfection. So once you have a virus ready, so and then you can do it for most experiments in the published work. We do the retroorbital injection, so allow us to do the system. Basically, this is a way of system administration, so allow to infect the brains, potentially look at the peripheral infection as well. And we inject after you inject the retroorbital into a mouse, we wait for about in the most cases 4 weeks, right, to look at the expression intensities, labeling patterns, and we could do we perform the detailed ex vivo and in vivo characterizations, and then it turned out the enhancer AAV with the CRE 89763 has shown the high efficiency and of the selective transduction of the brain BECs in mouse brain. And I also would like to say a couple words about the CRE sequencing. This is from a mouse CRE sequencing, enhancer sequencing we obtained from Catalyst, and then also learn blasts to compare with other species, including rat, human, and some other non-human primates genomes. And you can see that the CRE sequencing, enhancer sequencing is showing a high degree of conservation. So this indicates or suggests that the CRE sequencing will have a cross-species application potential. So in the next few slides, I'm going to tell you about our experimental details. And as shown here in these slides, our data indicate that the BEC enhancer AAV confers the brain endothelial cell specifically targeted following retroorbital injections in young adult mice. So we use the AAV PHEB capsid and the viral backbones, including BEC enhancer with a minimum beta globin promoter, and the payload is ACYFP2, the urofluorescent proteins. So I call in here we just said BEC enhancer AAVs. And the slide is showing some example cortical coronal mouse brain sections, and as you can see from the anterior to more posterior, and we normally will look over the brain sections at different, basically through the different positions. And turn out that if you look at the generally, you can see that it's pretty well. The generally it looks like we're only seeing the macrovessel structure labeled without seeing neuronal label. So essentially, it's a little neuronal label or other cell type label. And then when we look at the different brain regions, across different regions, you can clearly see a nice kind of microvessel like locus scale about 100 microns. So really, these enhanced AAV target microvessels, including capillaries. So initial round looks very good, right? And then we'll also we also would like to say, well, is this the gene expression labeling or gene expression label is driven by BEC enhancer or not? So what do we do? That we basically took away the enhancer, right? So this AAV is the same capsid with a minimum promoter, but without enhancer AAV. So we call it BEC minus AAV. In comparison, and this AAV really does not do much. Even with some immunostainings, you may see some of the sparse label, the neuronal labeling. We did not see any of the vessel structure. So that's indicated the enhancer, the BEC enhancer is crucial in terms of the gene targeting of the BECs. And in addition, also do the control experiment, just injected AAV PHEB with universal promoter like CAG with for the mitochondria. And so this is basic control experiment. You can see that result is although you use same AAV PHEB capsid, but without the BEC enhancers, now this the virus the labeling all sorts of neurons and glial cells, but it seems does not effectively label any blood vessels. And we would like to, so far we've seen quality is basically so far the real like initial experiment. We just do some of the mostly the qualitative observations. And in the next, we perform further rigorous experiments and validate the BEC enhancers labeling. And there's so the BEC enhanced after the AAV labeling, we verify their staining label and staining patterns using either use the mouse transgenic label. So we basically injected those AAVs into the Type 2 mice, or we run the mouse sections count staining with CD31 immunostaining. So this. And so Type 2 mice real labeled real labelized endothelial cells, and CD31 immunostainings really support verify the endothelial label because as one of the immunomarkers against the BECs. And so I should mention that although people that in the field, people have some of the endothelial labeling, but the endothelial cells not like neurons or some other or some glial cells. You can stain them. You can look at some clearly demark the cell body or the things. But endothelial is more like sometimes more like a sheet. Although you may see some of the that's punctate of endothelial nucleus, but the quantifying endothelial label can be difficult. And for our studies, we would like to do a rigorous quantitative analysis. So our engineering teams develop new approaches. And new approach to quantify the brain vasculature targeting. So what we did, I shown up here, is that we have those viral labeled images or immunostaining images or mouse genetic label images. So what the first we do that we would like we use our image processing tools to create the vessel masks. Right, so you basically segment the vessel and get masks and from different labels. And then you adopt some quantitative metrics using the DICE, precision, or recall indexes. So DICE index, as shown up here, you can read more details in our published paper. So DICE is really the DICE index. The numbers range from 0 to 1. 0 means the basically totally identical. So high numbers will usually mean two sets of the labeling showing high similarity. Precision is really means really the index showing the labeling specificity compared to sets of the mass, two sets of the labelings. And recalls we look at the efficiency or the coverage of the labeling by the specific tools. So here showing some of the nice quantification data of the BEC labeling by using our enhanced AAVs. And notice that we have a lot of experiment done. We injected those BC enhanced AAVs into the type 2 ANM mice. So type 2 ANM, those are genetic labeled vessels are labeled shown in pink, and AAV shown in green, and sometimes also corresponding with CD31 immunostaining. And let's look at the quantitative analysis shown here. Let's look at Dice index. So if we look at the type 2 versus the L2, they're shown about a range about 0.6, right? And on the other hand, if you compare with the BCAAV with either genetic label or immunostaining label, they're quite close. It's around about 0.6. Right. So that's showing that the enhanced AAV shown pretty good efficiency, And then we can look at precision and recall. They show, pretty much similar to the genetic label or the immunostaining pattern. And in addition to to look at 2D brain section categorizations, we're also using our light sheet imaging resource to do further categorizations. And these movies really shows the brain-wide vasculature label. And really show us there's a high degree of overlap between the BC enhanced AAV label and the type 2 ARN genetic label. All right, so the AAV label shown in green, and type 2 ARN label, genetic label shown pink, as overlaps in the emerge appearing white. And so this 3D imaging, you can see that there's a high overlap, and also there's the AAVs have extensive brain-wide labeling. You have a relative range of the microvessels. You have some of the capillary, smallest capillary cells, capillary vessels, also relatively large micro vessels. And so there's a note say that each grade is about 1. There's a grade showing the scale of each grade. Okay, so essentially they're showing the way a very intensive brain-wide labeling by BEC enhanced AAV. And so we move on. We also would like to address, we ask ourselves, does the BEC enhancer work with different capsids? Right? So as I told you so far, we use the AAV PHEB package. Those viruses use AAV PHEB capsid. Right? And the AAVBR was the first AAV capsid reported to be selected for labeling the brain-induced cells. And then we have replicated so we obtained the materials AAV PHEB, make it a CAG and EJP, and we injected this virus into the type 2 ANM mice. As you can see that, yes, the BR1 does label some about the vessels, vasculatures, but there are a lot of leakage for them, particularly in those regions of interest. We can label a lot of neuronal-like structures. And on the other hand, so now we use the capsid where we pack our virus into the AAV using the AAV-BR capsid with the backbone, the BEC enhancer backbone. Now things are quite different in contrast to the lots of leakage to the neuronal label of the BR1 CAG virus labeling. Now you look here, you can create, so you can look at real see those mostly the capillary labeling using the using the BEC enhancer with the AAV BR1 capsid. So this is a way, so this allow to conclude that the BEC enhancer improves AAV BR1 targeting of brain industrial cells. And if you look at those quantifications shown at the bottom, also confirmed our visually qualitative observations. And we went further. We characterized our enhanced AAV vectors in vivo and extending their applications to the live brain images. And this is an interesting experiment. So what first we do that we co-inject the AAV. So one the AAV enhanced AAV to label the brain vasculature with the AAV8P3 albumin scarlet cells. So they target the liver cells to produce the fluorescent target, the albumin. And then after a few weeks of incubation, so we can use two-photon microscope to make it a craniotomy and put the cortical windows. So we can image the vascular label as well as the blood flows in the live mouse brain. As shown up in some examples shown here, because the AAV8 produce M-scarlet labeled albumin, we actually be able to image the blood flow, basically because the blood plasma is labeled by albumin, right? And so you can see the flow here, and then the two-photon microscope also allow to simultaneously visualize the BEC labeling. So you can clearly see the flow is within the boundary, within outline, basically in the microvessels outlined by the enhanced AAV labeled in the serial. And you can tell from that you have labeling different size of the blood vessels in different field of view. So this you can see relatively large microvessel. And here we can already see in the individual capillaries. And in the real-time movie, you actually see that individual RBCs cause these capillaries. So this I think it's so clearly that both ex vivo and in vivo characterization support enhanced AAV can be enhanced with a successful tool can be used can have both in ex vivo in vivo application. And also note that there's little off-target label in peripheral tissues by BEC enhanced AAV through the retroorbital injections, which is systematic administration. And you can show here we look at liver, kidney, lungs, the little labeling with the enhanced AAV with payload SYB2. And even we use payload in ANMIs, you still see not much little signals. And so in the next, we also further look at to see if the enhanced AAV the administration route versus its labeling specificity and efficiency. This actually is important because for different applications, scientists use different administration routes. The top row shows the retroorbital injections of the enhanced AAV into the type 2 Cre-ANM mouse. So this is typical control. You can see a quite extensive, highly efficiency and selective labeling. Notice that some vessels actually some vessels are not labeling by the BEC enhanced AAVs, showing the purple, showing the pink here. So as for the intracranial injection, if you use the similar title we use for retroorbital, so you inject it, you see a lot of local infection, and basically essentially you don't see the specificities. And it looks like this high high titer viral injections, intracranial injection, you don't see. So BC enhanced AAV really lose the specificities. And and as for the intracerebral ventricular injection, ICV injections, and they are okay, but they don't spread too much. The labeling efficiency seems much low. Although they still retain the specificities, but they see a lot more vasculatures is not labeled. So this is important. So that the data support the BEC labeling or enhanced AAV labeling specificity and efficiency depends on its administration route. And so in order to, so yes, so I can show that now the BEC enhanced AAV can gain the gene access, right? So in access also we like to replace the fluorescent report with the Cre recombinase. This is important because Cre is very useful toward the genome cell type specific genome editing. So for initial round, we just replace the fluorescent protein payload SYV2 with the regulated Cre recombinase. And once we package virus and we inject a retrorbitally at different dose, right, shown here, high dose E11, E10, 9, and 8. It's kind of interesting to see that the high dose at this use, because we know and we inject it into we use the AI9 mice reporter mouse lines. We know that the Cre-dependent reporter mouse line is quite sensitive to Cre-mediate recombination. Right? And the high dose, you should see, you really don't see the much will be no specific BEC label. And I should note that here the T-cell middle signals are coded, pseudocoded in green. I actually see a lot of neuronal labeling, not so much of the BC labeling. This through the high dose of retroorbital injection of the BC Cre virus. And with the reduced virus, you can see preferential BC label, but still a lot of leaky. You can see leaky in the neuronal. So that turned out to say that this with the BEC enhanced AAV with regular Cre recombinase did not work well. And so the team is basically modified these payloads. And so what we did is to decrease the non-specific targeting of the BEC enhanced AAV using regular Cre. We actually improve these vectors by by introduce a couple of modification. And since the regular Cre is quite sensitive, so what we do is that we use attenuated Cre. So we just use those mutant Cre and to reduce its sensitivity. This help, and then we introduce microRNA targeting sequencing. And so this microRNA sequencing can trigger the transgenes degradation specific in neurons. So now we put this improved Cre payload, attenuated Cre with microRNA targeting sequencing, and now we inject this improved Cre AAV, enhanced AAV into AI9 mice. So you can clearly see it work much better. I should say very well in this case. So even we had the highest dose, like E11s in vivo retroorbital injections, you can clearly see a lot of labeling of the endothelial cells without leaky label of neuron or other cell types. And we also did this quantitative analysis also support our conclusion that we can use these new viral vectors for the gene access and labeling using the improved Cre expressions. To support to support the future potential applications in treating the brain disorders, particularly Alzheimer's disease. And then we carry experiments of the enhanced AAVs in aged Alzheimer's disease mouse model mice. So shown here, we injected first we injected the regular enhanced AAV with SYLP reporters. In this case, we done in virtue a older Alzheimer's disease mouse model, 5XCD mouse. This we've been using. This is about more than 1 year old mouse. You can see that a lot of histology section is stained by the anti-A beta, the lot of beta amyloid depositions. But still, with the enhanced AAV label pretty well. Really, the label is exclusive to the brain endothelial or microvessels, even in the highly AD pathologic brain regions, including cerebral the brain region. And in addition, we also injected the enhanced AAV expressed improved the Cree recombinase into 5XAD crossed with ANR report mice. And the left just shown one section, right? And so on the right, you show some high the zoom in field view of different brain regions. And so this even in these highly pathological brain regions, the labeling or the Cree recombination in the brain microvessels show up very well. Okay. So in conclusions, so we have generated BEC targeting enhanced AAV vectors. And the BEC enhanced element confirms targeting selectivities with different AAV capsids, including AAV PHEB and AAV BR1. And also I show that the BEC enhanced AAV expressing and improved Cree recombinase allow for precise genetic access and labeling. And based upon our preliminary work using the AD mouse models, our BEC enhanced AAVs have a potential therapeutic applications in treating brain disorders, including Alzheimer's disease. And so I would like to for this BEC enhanced AAV work, and the team, the group of picture of the team's members are shown here, and Dr. Eric Legacy Rivera is really the major leaders for this work, and specifically this work is made possible by the support of Brain Initiative Ontario project. And in addition, I would like to acknowledge other team members' help, and also would like to thank our collaborators, particularly Dr. Jonas Ting at the Allen Institute, and Dr. Bingling, who used to be at UCSD. And again, I would like to acknowledge the NIH grants funding support. With this, I'll be happy to take your question.
Thank you, Xiangming. Let's take some questions from the audience. So first, what are the main advantages of using enhancer AAVs over traditional promoter-based AAVs for brain cell targeting?
Hi, you can hear me well?
I can hear you and I can see you.
Excellent. This is an important question, and I think I touched upon this in my talk. But let me highlight a few key advantages of using the enhanced AAV compared to the traditional, the promoter-based AAVs. in terms of the brain cell type specific targeting. All right, so I should say that there's several, and the first one, as I discussed in my talk, I think that enhanced AAV allow for greater precision and specificity. So at one point in my talk, I mentioned that there's many more more of the a magnitude or more of the cell type specific or subtype specific enhanced element compared to broadly active promoters. So this suggests that enhanced element is a good way to go for if you're more interested in great precision in targeting specific cell types. And the second advantage, as I can see, that is enhanced element are very cool, right? The enhancers they sometimes their activity regulate. They sometimes enhancers are also region specific. So by leveraging those enhanced elements, we actually can achieve the contact and the state selectivity. So for example, we may come up with enhanced AAV targeting specifically developmentally activate those cell types. So I think these are some things where I'm quite excited about.
Fantastic.
Yeah. And then in terms, then another advantages, as we all know, the enhanced element is very short, a few hundred EPs, so which free up space in AAV vector backbones, so allow us to more rooms for payload, right? And also allow for more design flexibility. So this is a few, several key advantages.
Great. And could this approach be adapted to target other blood-brain barrier cell types or peripheral endothelial cells?
Yes. So I think the principle is the same. And there's really has a lot of growing single cell mapping, particularly in terms of multiomics. Right? So normally we can identify potential enhanced element, cell type specific element based upon ATAC data as well as the maturation data.
Mm-hmm.
So as far as you can, yes, there's some of the ongoing work that we would like to identify other vascular related cell type, in addition to BCs in the brain. And for peripheral, this is something I think is it's kind of nice. You can see that when we apply our BC direct enhanced AAV. There's little labeling in peripheral when we apply these to the inject these into retroorbital through the system administration. So yes, so this is nice to see this specific. It's only for mostly the transductions in brain versus the peripheral. And on the other hand, I believe we should be able to make peripheral tissue specific vascular targeting against AAVs.
And then what kind of therapeutic applications beyond Alzheimer's disease could then you target?
Okay, so this is a very forward-looking question, and so enhanced AAVs are still pretty new. Right? We're in early phase of development. But I should say that enhanced AAVs do have substantial potentials in terms of a range of applications beyond, the Alzheimer's disease. Right? So anything if those conditions such as traumatic brain injury, stroke, or some of the other disease, especially impact the brain-blood barrier integrities. For example, as I show you in my talk, so this is done in mouse models. We inject virus, and you can see that these enhanced BC enhanced AAV can transduce into serious cells even in the highly pathologic brain regions. They still infect pretty well. So in that case, if we can deliver those therapeutic agent to restore the integrity of the BBBs, definitely will improve the brain function or brain physiological condition. So this is generally applied to other brain disease beyond the Alzheimer's disease.
Great. And what's some of the biggest challenges in translating BEC enhancer AAV technology from mouse models to human applications?
I should say that humans are quite different. So a lot of a lot of talk even in today's symposium, people talking about AAV. So reality is, a majority of AAVs produced, including, we offered at our UCI Center for Neuroscience Mapping, right? So this is really for the basic research. And I also back to the enhanced AAVs, they're still in early stage, right? So a lot of them for now we're really doing the preclinical work, demonstrate the proof of concept, right? And the human, I think safety is quite important. So far we needed to do still a lot of work to do. Validate the specific efficiency as well as safety. Right? So there's still a lot of work to do before we get into the human. And on the other hand, personally, I think AAV can be used for certain human genetic therapy, but I think systematic delivery might be huge issue. Okay.
And what kind of biosafety considerations are associated with using AAV-based methods?
So AAV, so this enhancer is, I think, under generally those is BSL1, level 1 safety, biosafety level 1 is generally is quite. So enhanced AAV is in terms of packaging. The clone is quite very similar to traditional AAV to promote AAV we can use. Right? But now that we have much more choice. They have much more precisions by using enhanced cell type specific enhanced elements. Right? So in terms of safety, safety wise, that's why a lot of people like to use AAV because there's there be a level 1 safety requirement.
Great. And the last question, how long does the BEC enhancer AAV continue to express in brain endothelial cells?
Yeah, that's very interesting. I think Eric, the senior scientist led the work presented here. So the summer mice, we leave them for about more than a year. They're still, I think the labeling is still pretty good, right? So that's kind of interesting because this also one of the questions that relate to the cell state, right? Disease BECs renew, how soon they get a renewal and other things. Or another thing that, this is something, overall, so far, I think do see that, but in our experience, these enhanced AAVs, that it has long-term expression potentials in our hand.
Fantastic. Well, thank you, Zhangming, for your presentation and answering all our questions today.
Thank you. Bye-bye.
Bye-bye. So now let me introduce our final speaker. Ronette Mazur is a principal investigator and laboratory chief in the Gene Transfer Immunogenicity Branch at the Center for Biologics Evaluation and Research of the U.S. Food and Drug Administration. She leads a multidisciplinary research team focused on evaluating and mitigating the immunogenicity of gene therapy products and serves as a primary chemistry, manufacturing, and controls reviewer and biologics license application chair for first-in-class gene therapy products. Mazur's scientific contributions include high-impact publications, patents, and several FDA awards, including the CBER Center Director Awards. Her research encompasses T-cell immunology, algorithm-based HLA binding predictions, investigation of sex-based immunological differences in gene therapy outcomes, and development of tolerogenic approaches to enhance therapeutic efficacy. Prior to joining the FDA, she conducted research at the National Cancer Institute, developing immune-tolerant strategies for cancer immunotoxins, and served as the research lead at AstraZeneca, focusing on immunogenicity prediction for therapeutic proteins. Mazar holds a PhD in molecular immunology from Tel Aviv University through the NIH Graduate Partnership Program and completed her postdoctoral training at the National Cancer Institute. All right, let's make sure your slides are up and running. Okay, Ronit, take it away.
Good morning, everyone. Thank you for joining us today at the Scientist Cell and Gene Therapy Symposium. My name is Lonit Mazor from the FDA Office of Gene Therapies, and I'm excited to share with you our recent exciting research. Today, I'll be presenting our findings on the impact of biological sex on AAV immunogenicity. Research that reveals critical sex-based differences that we believe every gene therapy developer and clinicians should know about. This work represents a collaboration effort supported by the FDA Office of Women's Health, and I'm excited to walk you through our recent study. So not a lot of people know about the research in the FDA. The Center for Biologics has a strong and productive research program focused on mission-relevant scientific questions. The Mazor Lab is dedicated to developing advanced analytical tools and methods to support, mitigate, and improve gene therapy products, with a particular focus on AAV vectors. Our work spans from basic immunological mechanisms to practical regulatory applications, always with the goal of making gene therapies safer and more effective for patients. Today's presentation will take us through 5 key areas. We'll begin with AAV regulatory milestones to set the context, then dive into the immunological challenges we face with AAV delivery. From there, we'll explore what we know about immunological sex differences in response to viruses and vaccines, before presenting our findings on sex differences in pre-existing antibodies to AAV gene therapy. Finally, we will examine the mechanistic basis of these differences through our ex vivo studies. So we'll start with a short introduction about AAV gene therapy. Considering the audience, this will be relatively brief. AAVs were discovered in 1965 as a contaminant for adenovirus preps. It is a single-stranded DNA-powered virus, which is non-pathogenic in nature, and it requires a helper virus for replication, such as adenovirus or herpes simplex virus. It has a relatively simple protein structure that is composed of 60 subunits with overlapping sequences that come together to form the capsid, as you see here. And the capsid composes the majority part of the virus's weight. The genome of the AAV, of the natural AAV, is composed of 2 ITRs in both sides that are surrounded around a rep and a cap gene. For viral genome, vector genome design, the rep and the cap gene are replaced with the gene of choice, as well as other regulatory elements such as a promoter and polyadenylation signal and others. AAV has become very popular as a vehicle to deliver DNA for gene therapy due to several properties. First, it's simple. Its DNA is relatively easy to manipulate. Importantly, it has episomal expression in the host cell nucleus, which reduces the risk for insertional mutagenesis. And also, it has the potential, it has been shown to persist in non-dividing cells, allowing for sustained expression for many years. The regulatory landscape of AAV products has been rapidly evolving. Regulatory milestones start with the first submission here of the AAV product to the FDA in 1995. It was an AAV that targets the CFTR gene. Fast forward 22 years later, the first BLA approval by the FDA was for Luxturna in 2017, and it was followed by the approval for Zolgensma in 2019, representing the first gene therapy delivered systemically to a pediatric population. Then in the years 2022 and to 2024, we had 5 BLA approvals for Hemgenix, Elevidys, Roctavian, Beqvez, and Capillity. We're seeing an increasing rate of BLA approvals, which reflect both the maturation of the field and the pressing medical need these therapies address. Looking at the number of INDs submitted to our office in the past 30 years here, starting with the first submission that I mentioned earlier for the CFTR in 2015, we see a consistent growth in IND submission for AAV products with more than 40 submissions in the last data point in 2024. What particularly is striking is the timeline from IND to BLA approval, shown here on the right, which typically spans 5 to 10 years. This means that based on the current IND submission and the length for a BLA, we anticipate a significant surge in licensing applications over the next few years. Therefore, the FDA has a strong interest in having a better understanding of the factors that affect safety and efficacy for this technology. The therapeutic landscape of AAV therapy is expanding fast. As Reid and colleagues summarized in their 2024 review, clinical trials for AAV gene therapy focus mostly on neurology and neuromuscular indications, followed by ophthalmology and metabolic indications. Importantly, we're moving beyond traditional monogenic disorders into indications where females are represented equally, such as ophthalmology and cardiovascular issues, but we are even seeing indications where females are disproportionately overrepresented, such as neurodegenerative indications. This means that we are increasingly treating patient population where sex differences in the immune response could have major clinical implications. Looking at the AAV serotype of choice in different clinical programs, AAV9 is still the most utilized serotype, followed by AAV8 and AAV2. There is a large fraction of undisclosed or other, which includes non-natural AAVs or engineered vectors. Moving to immunogenicity. Due to its viral origin, it's not surprising that AAV vectors have some immunological challenges. Before the therapy, pre-existing antibodies that originate from the environmental exposure can neutralize the vector or induce accelerated clearance. These antibodies are prevalent in 30 to 85% of the population, depending on the AAV you use or the study or the methods you use to measure it. These antibodies can neutralize the AAV or cause accelerated clearance, leading to patients' inclusion in many trials. Exclusion of patients is bad because many of these indications don't have any alternative therapies, and the gene therapy is the only option those patients have. In patients that do have pre-existing antibody and were not excluded in some trials, these may introduce toxicity concerns, particularly during the dose escalation step, where patients with pre-existing antibodies can tolerate higher doses, which can induce toxicities in other patients that do not have pre-existing antibodies that receive the same dose or higher doses. Even if we do not see pre-existing antibodies, all patients are bound to mount a complex immune response after therapy. These immune responses are composed of innate and immune system activation, potentially rare infusion-related reaction, and formation of immune complexes. This dictates patient monitoring, and in many cases, sponsors choose to co-treat the patients with immune-suppressing steroids, which have their own line of adverse events and toxicities. days and weeks after therapy, we continue to see adaptive immune response against the AAV. This includes formation of cytotoxic T cells that can kill the transfected cells, which can lead to liver toxicity as well as other immune-mediated toxicities. Additionally, 100% of the patients go through seroconversion, which prevents further therapy, essentially limiting this therapy to a single shot. The factors contributing to immunogenicity, as described in the FDA's 2013 guidance for immunogenicity assessment of therapeutic proteins, and also the 2022 guidance specifically for neurodegenerative disease, describe patient, product-related, and treatment mode-related factors. Patient-related factors include prior exposure to the natural virus or the gene therapy. The immune state of the patient, for example, patients that are immune compromised have lower immune responses than hyperreactive patients. The genetic background of the patient, such as the HLA or the cream status, which refers to how foreign the vector or the transgene are from the patient's natural DNA. The sex, age, and geography, which we try to keep into consideration in our study that I'm presenting today, as well as other medication that the patient may take that can enhance or reduce the immune responses. Factors related to the product include epitope content, the aggregation, post-translational modification, for example, deamidation. We published a study in 2022 that shows that deamidation that spontaneously occurs during AAV storage can result in increased immunogenicity in some individuals that have specific HLA. Other product-related factors include impurities such as the host protein or the host DNA, the CpG content that activates the toll-like receptor, and empty capsid content that can increase the capsid antigenic load. Under treatment-related modes, we have the frequency, the dose, duration, the route, tropism, and combination therapy with other drugs all can lead to differences in the immune response. Another thing to consider is that these gene therapies include a vector and a transgene. Every transgene has its own immunogenicity risks, starting with the transgene that may or may not appear foreign to the patient immune system, and continuing with the risk of epitope spreading and mounting an immune response against the transgene, which could result not only in transgene loss and loss of efficacy, but even in finishing the treatment worse from where we started. So now I want to switch to discuss sex differences in the immune response. To understand the potential significance of sex differences in AAV response, let's consider what we've learned from COVID-19. Many studies show common observation that sex has an impact on the immune response to COVID-19 viral infection. The study here on the right that was published by Scully et al. Here we go. Compared the fatality rate in COVID-19 by male in blue and females in red. They found that in all countries examined, males had a higher fatality rate. In fact, males had 1.7 times higher death rate globally across the 38 countries in the study. And that was true for all age group above 30 years old, with male sex identified as a risk factor for death and ICU administration. Conversely, more females reported adverse events after COVID vaccines. And this pattern of male expressing and experiencing more severe diseases, but females showing stronger immune responses to the interventions, provide an important context for understanding potential sex differences in gene therapy response. The pattern I showed for COVID-19 is not unique. Sex differences have been reported in responses to numerous vaccines, including the DTaP, hepatitis A and B, HPV, MMR, influenza, even rabies vaccines, and more. The mechanism underlying these differences is complex, and the different cellular factors are interconnected. Cell differences affect viral entry and immune sensing through X-linked genes, such as the ACE2 or TLR7, along with androgen regulation of entry proteins. Females consistently mount stronger antibody and interferon responses, but this comes with an increased risk for autoreactivity. In vivo and human studies investigating the factors contributing to the immune differences include the effect of sex hormones, immune critical genes such as the CD40 or the TLR7 that are on the X chromosome, as well as microRNA, and of course the microbiota, which is known to be significantly different between males and females. All of these factors have been documented to induce critical differences in many functions of the immune system. And this biological reality led us to hypothesize that similar patterns might exist for gene therapy products. So this brings us to our central hypothesis. The stronger immune responses consistently observed in females might have a significant downside by boosting pre-existing antibodies against AAV gene therapies, potentially limiting females' access to this critical treatment. The image I'm showing here on the left is of Evelyn. She was among the first 15 patients with spinal muscular atrophy that were treated with Zolgensma. In the article in Science magazine, she was shown running here at 3 years old, when usually these patients do not survive past the age of 2. Based on the successes and promises of Zolgensma, we focused our study on AAV9, the AAV vector that was used to deliver DNA in this therapy. But we later expanded it to additional AAV to cover a broader sponsor population. So we are back to the slides that I showed earlier, but we added here another step, another part, focusing, showing the regulatory patient population that was used, that was evaluated during these clinical trials. As you can see, more than 50% of the FDA-approved AAV products are for X-linked indication, and therefore were not tested in females. The 3 approved products that are non-X-linked indications are targeted to either very young pediatrics, like here, or were targeted to the eye, which is an immune-privileged organ and may not indicate the sex differences as well. So we do not have sufficient and publicly available clinical data to address the question if gene therapy works differently in males and females. So to test our hypothesis, we designed a comprehensive evaluation of pre-existing immunity to AAV in male and female adult donors. We started with collecting serum samples from untreated adults initially and tested these samples for seroprevalence and titers to AAV9. Once we observed the differences, we wanted to confirm our observations using a larger second cohort from different geographic regions. Considering that the very diverse AAV vectors that our sponsors use for different indications, we next confirmed that our observations were true in 4 more AAV serotypes. Also, considering that binding antibodies and titers do not always have neutralizing activity, we tested AAV neutralization by the 2 cohorts. And lastly, we are starting now to investigate the specific cellular mechanism that may lead to these differences. So we established 2 independent cohorts to ensure the reliability of our findings. Cohort 1 comes from NIH Blood Bank in Bethesda, Maryland, serving as our initial observation group. We had 61 males and 37 females. Cohort 2, collected from Florida, Tennessee, and Pennsylvania, served as our confirmation cohort with 80 males and 80 females. The demographic were well matched between the sexes, with mean age of 40 to 50 years old and similar age ranges. In addition, both cohorts showed a same pattern in anti-AAV 9 titers. Females showed higher titers than male, and I will spend the coming few slides to describe those findings and how we found it in our interpretation. So to compare the seroprevalence and antibody titer, we use a direct ELISA approach with some modifications required for lab-made AAV products. Each serum sample was tested twice in 2 replicas and then repeated one more time. One sample was used in a plate that was coated with AAV, and the other was used in a plate that was coated with debris that comes out during the AAV manufacturing. We call this debris sham, and this negative sham control reduces the background and increases the sensitivity in lab-made assays and research-grade reagents. The pre-existing antibodies in the serum are represented by the pink antibodies, and if those antibodies are present in the serum, we detect those with anti-human antibody that is labeled and can give a color reaction. The results were analyzed in 2 ways, as shown in this example here on the right. To calculate the serum, we calculate the highest dilution, where we can still see a signal, after subtraction of the sham signal, of course. So in this example, the signal goes away between 10,000 and 100,000 dilution, and the computer calculates an interpolation of around 87,000 dilution factor. That would be the titer. The other analysis looked at signal-to-noise ratio at the lowest dilution. Each of these analyses has advantages and challenges, with higher accuracy with the titer calculation and with a significant advantage of reduction in critical reagents in the signal-to-noise approach. We try to use both approaches in most of our study. So this is probably the most striking observation and the most important slide. Our results revealed a striking pattern. While males and females show similar seroprevalence rate, here as indicated by the gray boxes on the bottom of the graph, we observed that within the seropositive populations, significantly more females had high titers, as you can see here and here, while male had more of the lower titers. These findings suggest that while the exposure rate may be similar between male and female, the immunological response to that exposure is significantly different. The statistical analysis confirms what we observed qualitatively. Female serum consistently showed higher titers in pre-existing antibodies against AAV9 compared to male. This difference was statistically significant and consistent across both cohorts, eliminating concerns about chance finding or cohort-specific effect. This has important implication for clinical practice, as many gene therapy protocols use titer cutoffs for patient eligibility. If adult females systemically produce higher titers, they may face disproportionate exclusion from these life-saving therapies. Moving beyond binding antibodies, we conducted a neutralization assay using a nanoluciferase reporter system. In this assay, we transduce cells with AAV9 delivering a nanoluciferase gene. The transduced cells show a luminescent signal. If a serum sample containing neutralizing antibodies, this will prevent the transduction and reduce the luminescence signal. So here we show on the right 2 representative results. The right sample is seronegative. It's seronegative and is non-neutralizing. Therefore, the transduction efficacy is close to 100, regardless how much serum we add. The sample on the left is neutralizing. Adding serum inhibits the signal. As we dilute that serum further, we have fewer and fewer neutralizing antibodies, and the inhibition goes away. In this sample, we calculated around 1200 to be the dilution where we see 50% inhibition. This functional assay tells us not just whether antibodies can bind to AAV, but whether they can actually neutralize its therapeutic activity. So are the visual results for the neutralization of 60 randomly selected samples from cohort 2. Females showed strikingly higher neutralization activity against AAV compared to males, as indicated by these white large spaces. We also calculate the NC50, the dilution at which all seropositive samples in both cohort 1 and 2 induce 50% neutralization for the statistical analysis. This resulted in a statistically significant stronger neutralization in female. These findings have clinical implications for gene therapy trials, particularly in study design that includes seropositive patients. These differences in neutralizing activity could substantially influence therapeutic outcomes, with adult female patients potentially experiencing reduced treatment efficacy. Remarkably, when we performed a paired comparison of neutralizing activity, meaning we identify male and female samples from the same cohort with very similar binding titers and compared their neutralizing ability to those samples, we found that females showed somewhat stronger neutralizing activity even when compared to males with similar binding titer. So this suggests that it's not just the quantity of the antibody that differs between the sexes, it's also their functional quality. Female antibodies appear to be more effective in neutralizing AAV at higher dilutions, indicating either higher affinity binding or more efficient neutralization mechanism. And this functional difference could be even more clinically significant than absolute titer differences. Next, to determine whether this was an AAV-specific phenomena, we tested multiple natural AAV serotypes that are commonly used in clinical submissions, in regulatory submissions. The results were mostly consistent. Females show higher titers of pre-existing antibodies against most natural AAV serotypes, including AAV5, 8, and 9, showing significantly higher titers in females, and AAV1 and 2 that showed a near significant trend, but females were always higher. This broad pattern suggests that sex differences were observed is not vector specific, but rather represent a fundamental difference in how males and females respond immunologically to AAV exposure. The binding studies identified many seronegative samples to AAV9 or the other serotypes, which makes sense since some AAV serotypes are more prevalent in specific geographic regions. So we asked about overall seroprevalence to all 5 serotypes. We found that all donors were seropositive to at least 2 AAV serotypes, and most were seropositive for more than 3. Interestingly, while we saw sex differences in titer magnitude, we did not observe any sex differences with regards to the core prevalence pattern, which suggests that exposure patterns are similar between the sexes, but the immunological responses to that exposure differs significantly. Next, we performed a correlation analysis. This analysis revealed insight about the relationship between binding and neutralizing antibodies. We found that most binding antibodies were indeed neutralizing, but notably 20 to 35% of the binding antibodies were not neutralizing in our assay. We observed some correlation between titer measurement and signal-to-noise ratio, which is indicated with the different colors for each of the data points. But importantly, signal-to-noise ratio did not correlate as well as the binding work with the neutralizing activity. particularly missing many low to mid range indicated by the color of the neutralizing response. And this heightens the importance of functional neutralization assay as well as development of high quality ELISA assays for binding. Next, we asked about the immunoglobulin subtypes because the different subtypes have different effector functions, which may result in different immunological patterns and maybe explain the stronger neutralization in the paired samples. We found that IgG1 was the most prevalent isotype in both sexes, but IgG1 titers in the females was significantly higher than in males, which I think is not surprising considering it's a primary component of the total IgG. Interestingly, though, we found different subdominant subclasses patterns. Males showed IgG2 as the subdominant class, while females showed IgA. This IgA pattern in females is consistent with observation with other viruses. For example, female primates exhibited greater IgA antibody titers to simian immunodeficiency virus, suggesting this may be a conserved sex-based immunological difference. These subclasses differences could have important implications for antibody function and persistence, as different immunoglobulin subclasses have distinct effector function and different half-lives. Next, we aimed to try to understand what drives these differential responses. We conducted a cytokine and chemokine secretome analysis using human PBMCs that were stimulated with AAV9 and omics analysis. This approach helped us model the earliest immunological event that might explain the sex differences we observe later in the pre-existing antibody responses. The secretome array revealed innate activation after AAV9 stimulation, with notably different patterns between males and females, and several cytokines and chemokines that show differential response. So based on the secretome analysis, we dived deeper. The mechanistic analysis revealed that female PBMCs exhibit significantly stronger innate activation in response to AAV stimulation. Particularly, females had higher responses to CXCL10, CCL7, CCL5, and CXCL6 chemokines. As well, under the cytokines, we found differences that were striking for IL-6, GM-CSF, and IL-beta. String pathway analysis revealed that the responses help us identify an immunological signature between these sexes, and they suggested involvement of an innate migration pattern as well as innate inflammatory response. This provides us with a foundation for mechanistic studies that we are working on right now in the lab, trying to better understand the cells and components that have a role in this differential immune response. So let's summarize our key findings and the implications. Females consistently show notably higher baseline antibody titer and stronger neutralizing activity against various AAV serotypes. Our analysis of immunoglobulin subclasses revealed distinct sex-based differences, and female PBMCs displayed heightened inflammatory responses when stimulated with the AAV. The clinical implications are significant. This stronger immune response in females could lead to lower eligibility rates for gene therapy trials, especially among females of reproductive age. Moving forward, our research prioritized focus on uncovering the hormonal, genetic, and epigenetic mechanisms underlying these sex-based differences. And understanding these mechanisms will be crucial for developing strategies to optimize gene therapy access to all patients. So I want to thank you for your attention throughout this presentation, and a special acknowledgement to the heroic technicians and trainees in the FDA that have survived a lot of changes and demonstrate a lot of flexibility and professionality. Specifically, Stephanie Warrington and Leila Abdelhamid that did most of the work that I presented today. Other members of the lab, as well as our collaborators, particularly Trish Hogan and Morten Syrup from Promega, who developed the NanoLuc neutralizing assay. And of course, a great shout out to FDA Office of Women's Health that funded this research and made it possible. And again, thank you for your attention, and I'll be happy to take questions now.
Wonderful. Thank you, Ronan. Let's take some questions from the audience. So first off, what other factors may affect the level of pre-existing antibodies?
Okay, good question. We covered the sex differences, and we mentioned briefly geography. It's quite astonishing, the differences of different seroprevalence in different states within the US and also within the whole world. Importantly, there's the time since the last exposure, because our immune system and the antibody titer does wind down, as well as serotype in previous exposures, because we do see that there is a lot of cross-reactivity across the different serotypes. One more thing that I want to mention that can affect these factors is from the sponsor's side. So the way we design the assay, either measuring the titer or the neutralization. So how we look at the background, how we interpolate the curves and calculate the titer, those can also really affect the level of pre-existing that we can see.
Great. So in your presentation, you were showing that females have higher antibody titers, and given the recent clinical cases have linked antibody-mediated complement activation to serious adverse events, do you think females may be at a higher risk for these complement-related toxicities? And should sex-specific safety monitoring be considered?
Okay, so the short answer is yes. Referring to the complement mediated, so we see TMA cases, and we believe that they are mediated with complement and antibody mediated. So we don't have clinical data yet that connects the level of the pre-existing with the TMA and how it affects if it's going to happen or not. But I think it's important to try to collect this data in an organized manner and do this comparison. So that would be a valuable study to do.
Great. And your analysis for the immunoglobulin subclass showed females had higher IgA levels while males had higher IgG2. How are these different profiles affecting not just vector neutralization, but also complement activation and safety profiles in AAV therapies?
Yes. So the IgA is known to be involved in the alternate complement pathway, whereas the IgG usually are involved with the classical one. So indeed, some studies in the literature show that AAV toxicities were mediated by the alternate pathway. So it's possible that the high IgA in females may translate to differences in the specific toxicities. So going back to my previous comment, we need more clinical data from females at adult age to establish if there is such a discrepancy.
And what is your kind of next steps for your group in terms of maybe this research?
So I think I mentioned we're very interested in understanding the mechanism and really finding the specific cells that are involved in these differences, as well as we would like to take it and compare the toxicities that might be related to antibody-mediated responses.
Fantastic. And one final question. So that given that females show significantly higher pre-existing antibody titers against multiple AAV serotypes, how should this finding influence patient eligibility criteria in future AAV gene therapy trials?
Yeah, I think that's the question when people hear about the study that they ask a lot. So if you remember, the first observation was that seropositivity was not different between the males and the females. But the titers in the seropositive samples were higher in the females. So if a clinical trial excludes all seropositive subjects, that should not result in differences in the eligibility between the males and the females. But if the sponsor chooses to include subjects with very low titers, or their assay is not detecting all the seropositive samples, in that case, we may see more females being excluded. And that is something we are very interested in understanding.
Well, thank you so much for your wonderful presentation, Ronan.
Thank you.
So that concludes today's presentations. So tune in tomorrow for more presentations in our symposium. So on behalf of the scientists, I'd like to thank our whole panel for sharing your work. Bill Lager, Ajinkya Sase, Zhongming Zhu, and Ronit Mazur, and our symposium sponsors, Vector Biolabs, Shimatsu, Takara, Sino Biological, Beckman Coulter Life Sciences, Biorad, Taconic Biosciences, PHCBI, BPS Bioscience, AdGene, Innovative, and Mission Bio. If anyone has further questions, please consider reaching out to the speakers directly. Their contact was shown on the screen right after the presentation and is right now also on the screen. So as a reminder, the symposium will be archived on the scientist website, and you'll receive an email notifying you when the recording is available. Make sure to follow the scientists on social media for updates on future events. I'd like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. Thanks, everyone, and goodbye.